Hi, everyone. Thank you for attending. This is our part 2 webinar for the Innovation Across the Phenotypic Translational Divide webinar series. So just a few housekeeping items. So some webinar instructions, which hopefully everyone's familiar with using WebEx at this point. But everyone's muted upon entry, and you can unmute by selecting *6 on your phone or clicking the mic symbol next to your name or under audio connection on your computer. And you can also use a chat service to communicate with the host throughout the webinar and to ask questions to the host presenter or the attendees during the question period or even during the presentations. And so this is being recorded and we'll be starting now. So thank you all for attending. And the slides are available at this link, this bit.ly link, kf-webinar-2. And I've put a box at the top of many of the slides. So if you don't get the link right this moment, you can see it on some other slides too. So we invite you to upload the slides and follow along. And the agenda is linked here, and also the slides are also available on the website too. So, and we've emailed out the agenda to you as well. So hopefully you have all the information that you need. Oops. And I just want to give a thank you to the organizers, for everyone that's helped put together these webinar series, in particular Valerie, who's done a tremendous amount of work in getting this organized and bringing everybody together. Also Adam, Deanne, Allison, and Melissa. And with that, let's go on to do the introduction to the webinar, and I'm going to give it to Valerie, and I'll advance the slides for you, Valerie.
Perfect. You can go ahead to the next slide. Perfect. So Nicole said I do a lot of work, but the truth is all of you do all the work, and we do a lot of listening to you all, especially throughout the duration of the Kids First program. I think you've seen this slide before, and it's basically a summary of the results of our strategic planning exercise. It was actually strategic replanning. It was, we were about 3 or 4 years into Kids First at that point, and we wanted your input on what we should be thinking about as we go into these final years of the program. And believe it or not, we are wrapping up the 5th year of Kids First right now. But what was really important in this strategic replanning exercise is that all of you highlighted this need for really thinking about clinical and phenotypic data extraction, harmonization, and curation. At that time, we knew that phenotype harmonization across the wide diversity of data sets that you are all contributing and represented in the entirety of Kids First was going to be a challenge. But we also recognized that it was going to be really important, and especially this comes up when we're talking about functional validation with model organisms as well. Next slide. But as more data becomes available to the wider research community, to date we have 13 Kids First data sets out there. And you all are making discoveries. There are 18 publications, a lot of them that got published recently. So you all are on a roll right now. But as you guys start to make discoveries, we're starting to see even more opportunities in making these data sets even more stronger. And by stronger, I mean, more interoperable, more opportunities for working together. Next slide. And whether those opportunities are in finding overlapping phenotypes or in finding ways to capture and analyze a variety of phenotypes or data modalities, in many cases, some of these data types we didn't even think about at the beginning of Kids First when we were so focused on genomics. But during the Part 1 webinar held in April, listening to all of you was really helpful. You are all the experts in your respective fields, in your data sets. And it's been really helpful for us as NIH staff, and I think also members of the DRC who, is charged with facilitating this type of harmonization and understanding tangible examples of how we can make the Kids First data resource and the data sets that you contribute to it even better, even richer. Next slide. So with that, and it's related, that I really want to thank our speakers from the first webinar. All of you gave really excellent presentations. Like this one, it was a 3-hour webinar, but everyone said it went by quickly and it was scientifically interesting. And I'm sure that today we're going to build off of that success. I can't wait to listen to this next batch of investigators and to hear more of your thoughts in our brainstorming session today. And with that, I'll actually hand it back to Nicole.
Yes, so like our last webinar, our objectives today are to learn about clinical phenotyping efforts that are being done by all of you, the XO1 investigators, and also become familiar with some of the existing tools that you're using for your data collection for phenotypes and for data extraction and data harmonization. After we hear the talks, the lightning talks, we're going to have a brainstorming session again with some guided discussion questions where we can talk about and document some ideas on ways that we can improve our current methods. Networking isn't quite the same when you're virtual, but as much as we can, we are trying to network and get to know each other a little bit through the work that we're doing and through our discussions. So I'm going to turn it over to Deanne, and she's going to talk about some of the kids' first phenotyping evaluations that have been done.
All right, thank you. So without further ado, I'm going to jump in. Let me actually skip the first slide. I just want to briefly talk about this analysis. In the past, we've decided to look at the interoperability of phenotypes inside of Kids First, because that's part of Kids First mission, is to help study and look at the genetics and genomics of congenital heart defects and cancer. And of course, all of these come in different forms. So we were interested in looking at these analyses or the phenotype sets that came out of the different Kids First cohorts and try to determine how similar, how different, and how connected they are. So one way to do this is to start with a phenotypic tree, like an ontology, and this is a figure actually by Nicole that shows you how an ontology organizes its data. Most of you probably know that if you get further down the ontology tree, you go from more general, let's say phenotypic abnormality would be here at the top. And as you go down the tree, you get more and more specific until you get down to something that may be very clinically specific, like motor neuron atrophy or deeply set eyes or lack of smell, hyposmia, if I could say that right. And going up the tree, you get progressively more general. And as you get more general, you start getting more intersection of terms. The reason why this is interesting to the way we think about phenotypes is as you look through different cohorts, people have different ways of expressing a certain trait. You'll have people that may describe a child with abnormal eye morphology, but another clinician may come back and do something much more specific under abnormal eye morphology. So in order to integrate those two profiles from two different patients with different levels or precisions of definition, to do that, what would be helpful is to express something with a very specific ontology term through the tree up through its more less specific ontologies as a complete fingerprint. So for example, if someone has deeply set eyes, set up a fingerprint through this red path to the top. So let's say a deeply set eyes, you would have a deeply set eye and also a phenotypic abnormality, and you can't see the rest of the numbers one here, but there'd be a number one for each of these traits. And the same thing for each of these subjects. If they have a certain profile, anything else that they have as well. So it's not just one trait, it could be multiple traits, could be ones and zeros in a column. And those mathematically connect as vectors or binary vectors. And there are mathematical ways of establishing similarity between binary vectors, which I describe here. If you want more information on the method, you can go back to the slides. Pardon me, you can go back here to the methods and take a look, but I'll walk you through some of what we're doing. So when I characterize, this is work done with an intern last summer named Lilia Becker. We took the Kids First phenotype data dump that Bailey and Chris Friedman did for us. And we ended up characterizing every phenotype as one of these complex vectors as a characteristic across the entire child's phenotype or the individual's phenotype. And one of the things that you can do in the first pass and just looking at this giant matrix of ones and zeros where every row is an HPO phenotype term and every column is a patient or a subject, we noticed that the majority of the subjects in Kids First have about 10 HPO terms going up the tree in their fingerprint. So their profile has at least 10 different terms. Now, that might be because there are very few patients with very complex phenotypes, which we believe to be true. But there are some patients or subjects, I keep on saying patients, I'm sorry, I'm a chop, but there's a lot of subjects that have a large number of phenotypes, mostly because they have very complex phenotypes initially clinical phenotypes that had multiple layers. There's, some children could have, up to 20 different unique phenotypes or more. And therefore, when you walk up the tree, you start inheriting a lot of phenotype terms in their vector. So this is kind of looking at the diversity within each patient of what kind of phenotypes they have inside their fingerprint. You can also look at, this is a little bit more complex plot, but what it shows you is the number of HPO terms, this is the number of terms in the y-axis, and this is the subjects that have that term. So there seems to be, there are a lot of, a few HPO terms show up a lot, which we know because as you go up the tree, you get more and more specific, less and less specific. So many of these HPO terms are going to show up in high frequency. And they're going to be, so you'll have, but you also have a lot of patients with terms that are unique to them, which is out here more. So this is there are, actually I'm saying this wrong, but there are a number of rare phenotype HPO term. So this is a complex graph and I probably should have shown it. I need to set it up a little bit better, but we're essentially looking at the sensitivity of the HPO terms to the number of people with that term. This is a more interesting graph. This shows if I take the similarities between each of those fingerprints and think of it as a correlation graph. If you look at the upper side here, I have to move this. I'm not sure if you can see that window, but I'm going to move the window. At the upper right-hand side of the graph, we're looking at the upper right-hand side of a correlation matrix, and what this measures is the similarity from subject to subject. So each of these little tiny dots, there are 4,000 columns and 4,000 rows. And what you're seeing is a similarity from person to person. You're not seeing a diagonal, which should be completely dark, meaning that each person is 100% similar to themselves. I removed the diagonal, and we're just seeing the similarity of each individual to every other individual based on color code. So the darker the color, the more similar they are in their phenotypes. And we notice, for example, the greatest amount of similarity are in the oral facial clefts. And we think that is because they have relatively simple phenotypes and they repeat a lot. So this is no surprise that in the 3 cohorts that we have in Kids First, they're very similar. We also have a little bit more similarity, but some diversity in the congenital diaphragmatic hernias. And as you start looking at the lighter and lighter colors, you're seeing more and more diversity within cohorts. I should have probably moved this little yellow line right here a little bit further up. But what you're seeing is this is the childhood cancer and birth defects cohort. They're not very similar within the cohort because it's a very diverse cohort of children with birth defects and cancers taken from CHOPS archives, medical archives, and they're very diverse. We also see something similar in the CBTTC and the children pediatric brain tumor atlas, and we believe that's because these children were not necessarily ascertained under the same phenotype protocol. So we're not asking a very specific set of questions of these children. We're just taking what's in their medical records or additional information to their medical histories. And we see some of this kind of medium similarity in the PCGC congenital heart defects. They had about 160 terms that were phenotyped within those terms. Though they had some others, they were kind of carefully phenotyped in a system. So they're kind of self-similar in that way. But other things that pop out from this, what's really interesting isn't just the within cohort similarities, but also the ones that are between cohorts. For example, the congenital diaphragmatic hernias and associated congenital anomalies have a lot of similarity to the PCGC heart defects, and that's because there are a lot of children in the CDH data that have CHD characteristics, so that's where they overlap. But another one that I thought was really interesting is the hemangiomas. There's also similarity with the congenital heart defects, and that's because a lot of the children with hemangiomas, there's also some congenital heart defects. I'm not saying that these children have that, but there's precedent for a relationship between the two, and so forth and so on. So you can see oral facial clefts and syndromic cranial denervation disorders have some similar phenotypes across the entire cohort here in this big block. So this is really good at picking up similarities, but in a way it also is really good at picking up differences. So when you have a very pale color, it means there isn't a lot of overlap between the cohorts, at least in the presented phenotypes. Doesn't mean that there isn't an overlap technically in the cohorts, just that the phenotypes weren't presented to us that allows us to look at the differences. Next slide. I'm just going to show, this is kind of a numerical version of the earlier graph. The redder it is, the more similar within similarity the cohorts are. Off the diagonal again, the more blue it is, the less similar they are. So this is the cohorts that are very similar within each other are kind of self-referential, or possibly they were all phenotyped into the same system. So they may or may not interoperate well with others. I'm not saying they won't, but they have very limited phenotypes. So they may not spread out and do that cross-diagonal thing we were talking about earlier. But there's some similarities. Again, here's the this is the syndromic cranial dysnervation disorders. You can see that you probably can't see the pale pink colors right here, but you can certainly see it's about 0.25 average correlation. Here, I'm just showing the proportion of subjects with strict identical phenotype fingerprints between the cohorts, between cohorts. It's kind of similar to the previous slide, except this is strictly identical. So instead of looking at just like the general term of like the general similarity, we're looking at the strictly identical cohort. And you can, I'll skip over this slide, but you can read it, but it's essentially showing that kind of similar to what we saw in the earlier one, but we're also seeing that some of these cohorts along the diagonal don't have a lot of diversity within their own selves. So you're seeing a lot of strictly identical cohort phenotypes within these groups. So then if I remove, if I do a step where I actually remove all the similarities within these cohorts. So I'm going to remove anything that's more than 99%, or at least, or 0.99 cosine similarity. So things that are pretty much identical. A lot of the identical ones drop out, and I'm left with the most diverse set of samples. That doesn't mean that they aren't similar in some higher, some lower order, like 0.8 or 0.89. I completely just remove 0.99. And when I cluster these, you end up with these interesting groups of slightly similar, but not completely similar, but profiles of children along the diagonal. You can especially see it along the diagonal. No, we're no longer looking at cohort-specific kind of clustering, which you see here because I left the original ordering of the cohorts here. So this is, for example, the PCGC, the leftover CDH after we removed all the identical cohort, all the identical people. And when you do this, this is the kind of thing you'll see. Like if I zoom in, pardon me if I jump back again, but there's a little tiny square that you can barely see right here at the tip of this arrow, this little square right there. If I zoom in on that right here and I look at the individual patient IDs, you can start seeing why they're similar. So this is a very small subset of the infantile hemangiomas data set, but you can see why their phenotypes are similar. So it allows you to do kind of a selection. A lot of them have heart defects, not all of them, arterial stenosis, arterial tortuosity, and then of course facial hemangioma, and then some sort of cerebral vasculature anomaly. I'm not sure why this guy dropped in. He's kind of like the black sheep. I'm not sure why he dropped in, but there he is. But he's one of the ones right in the center, and somehow I think he's probably selected because there's enough similarity between the facial hemangioma and arterial stenosis. You can see they're both strong terms that he probably dropped in. So summary of what I've just done, selectivity matters. So the similarities I found are really affected by the deliberate design of how phenotypes were collected within each study. If they're not done agnostically, because they're most of us design our studies to ask specific questions. If they're not agnostically designed, we have a better chance of having a phenotype group or a group, your own cohort, separate itself from the other cohorts. So it makes it a little bit more difficult for you to interoperate with other cohorts without help from the other investigators in the X01, which is not necessarily a bad thing. Might be a good way to stimulate research. But more general phenotyping from family histories and data import, that a little bit more agnostic phenotyping might be less selective and designed, and maybe get rid of some of that batch effect that you get per cohort. And again, I've mentioned this before, and other people have too, but it would be great maybe to have a little bit of a test pilot for trying to do deeper cohort phenotyping. I know, for example, orofacial clefts possibly have deeper phenotyping data available. It's just that we need to put effort into that and try to integrate more of that data into kids first. But that's pretty much that for the phenotype. Now, do I have a couple more minutes, Valerie, to go through the survey results, or should we skip it?
Why don't you do it quick?
All right, real quick. So we did a survey back at the end of last year where we were looking at how people were involved with Kids First. This is a phenotyping survey. You can see most of us that responded were principal investigators. And the data that was typically collected was demographics, and then you can see a bunch of other stuff. It's really diverse in how people are collecting their data. For the most part, data was designed from a design clinical study, right? And some people collected from medical records. Both of those are really helpful. We were concerned about how our minimal phenotyping elements, if you're using the minimal phenotype elements and you haven't given us some feedback on that, we would love to, even outside of the questionnaire, we would love to hear from you. Some of the feedback is here, red is kind of like the more negative or critical stuff. The blue is more suggestive, like how we can change it. For example, treatment is a very difficult data element to acquire, and some projects can't do that. So these are the kinds of feedback that we were looking for. And the most pressing needs for phenotype help, coming back to the possible need for pilot projects for deeper phenotyping. You can see that this is the most pressing needs that people had. And if you want to go back to these slides and look at these questions, I'll show you in a second here. But responses, these are some of the feedback. Essentially, the first one pretty much sums it up. We already collect comprehensive phenotype data. We're not sure that the quality of data is available for some Kids First projects. We think there should be standards set for phenotype prior to receiving a award, and then just having specimens available isn't useful. So if you want to see the rest of this, you can look at I've hidden the slides, but if you go back to the slide deck, you can look at the rest of the results.
And with that, since I'm talking, I'll introduce Dr. Gharavi, who is the PI of the congenital anomalies of kidney and urinary tract defect cohort.
Thank you. Well, hi, everybody. Thanks for giving me the opportunity to talk about our work. We just got a bunch of sequencing done, and we're excited to analyze the data and move these projects forward. This work is done in collaboration with my colleague, Simon Sana-Kerki, and these are the folks who actually do the work. Hila and Miguel and Enrico are running these projects, and David and Sarath are doing the bioinformatics, and Madalena is doing a lot of data collection from different cohorts. Can I advance the slides, or do I ask to be forwarded? Oh, okay, thank you.
Nicole will do it for you.
Thank you. So for those of you who are not spending your time thinking about the kidney, on the left is a presentation of a survey of OMIM and other databases to see how many Mendelian disorders are associated with a kidney or urologic defect. And we did this cataloging a couple of years ago, and we identified 625 genes that are associated with any kind of kidney or urologic defect. Of those depicted here in blue, roughly around 300 are associated with congenital or developmental defects. Most of them are recessive, as you can see. So this tells you there's a wide spectrum of genetic disorders associated with congenital defects. There's a tremendous heterogeneity. And there's also tremendous heterogeneity in the presentation of these syndromes. On the right, you can see the spectrum of abnormalities you could see, which can involve one or both kidneys. It could involve the ureter, the tube that connects the kidney to the bladder, as well as malformations of the bladder themselves. There could be abnormalities in the backflow of urine from the bladder into the kidneys, causing vesicoureteric reflux. You can have duplications of some of the systems. You can have cyst formations. So there's a lot of variation that can occur, that these things can be bilateral, unilaterals, and importantly, they also occur in conjunction with other malformations elsewhere in the body. Next slide. So a number of years ago, when microarrays came online, we started systematically looking at our patient population to see whether we can find CNVs that are associated with disease. And to our surprise, we realized that this issue of heterogeneity really applies to CNVs as well. And now we've continued this work, and in a recent survey of about 3,000 cases, we identified a pathogenic CNV diagnostic of known genomic disorders in about 7 to 10% of cases. And so there were at least 40 different CNVs that are spread out across the landscape, the genomic landscape. And on the left, you see the slide depicting sort of one of the most common ones that we see. And it maybe doesn't project really well, but the symbols that you see next to the chromosomes are small kidneys and then a brain and a heart, just to drive home the point that these pathogenic CNVs that we identified have already been described for the most part in the context of neurodevelopmental defects or congenital heart disease. And the top loci that we identified are depicted here in the bottom: 1Q21, 4P, 16P11, 16P13, 17Q12, and 22Q11.2. So these accounts some around 60% of all congenital defects of the kidney. The first time we found these high load of CNVs, pathogenic CNVs, and reported this about 10 years ago, I think I got complete silence from the audience in a pediatric nephrology conference. But then since that time, I think there's been a lot more uptake, and more people are now using microarrays as first-line diagnostic tools for these congenital disorders. And you can see to the right that the diagnostic yield here summarized for CACUD is in the same range as what has been described for neurodevelopmental disorders, for cardiovascular malformations, and so on and so forth. So I think this is one important element. And a lot of these patients also have a number of novel CNVs that are not recurrent and may be pathogenic and deserve attention in some of them when they're very large or involves some interesting candidate genes. So CNV analysis has to be part of the analysis of these congenital defects. Next slide. We've also done exome sequencing, leading to identification of new genes for CACUD and congenital defects. One example of this in a case control association study, we identified this gene called GREB1L, which we found in patients with congenital kidney defects. But then subsequently, two other groups contemporaneously reported mutations in this gene, but ascertainments were really different. For example, one of them studies really looked at patients with severe fetal anomalies and fetal demise. And that's because the clinical features of these cases can be really also heterogeneous. There are uterine abnormalities, clinodactyly, missing roots. Congenital heart defects have also been described in these patients. And so this is a novel gene that was identified. We've also collaborated with other groups because of this heterogeneity. Many of us found some really interesting variants, but we're unable to confirm whether this is actually causal without having additional cases. And so we exchanged that data information, and particularly, for example, the next slide, we found, this is in collaboration with Friedhelm Hildebrand's group at Boston Children's, where he described mutations in this gene ZMYZ2 associated with, again, a spectrum of malformations. Most of the mutations here are de novo, suggesting that there's a real problem with reproduction in the setting of these mutations. But as you can see, there's a spectrum of phenotypic abnormalities associated with mutations in this gene as well. So this told us that we really do need to collaborate, but also really extend our phenotypic analysis of our patients to be able to get anywhere and maybe enhance our ability to collaborate. Next slide. So these are the phenotypes of the probands that we submitted for the EXO-1 project. There were 515 cases that were sequenced, and you could see that on the left, the spectrum of phenotypes involving the urogenital system, going from reflux to renal agenesis and so on and so forth. You can read on the right. But about 165 out of these 515 cases have additional extrarenal phenotypes that you could see: genital abnormalities, cardiac anomalies, neurologic anomalies such as epilepsy, abnormal brain structure, developmental delay are very common, and a bunch of other things. So we think that as we're trying to be more meticulous about collecting this information, we're getting these data sets from various collaborators around the world, as well as our own medical center. We're trying to be more systematic about highlighting these and using sort of the template also provided by the Gabriella Miller Fund, the Kids First project, to populate this information, so to increase our ability to collaborate. Next slide. And so the aims of our project is fairly simple, is to identify, the pathogenic CNVs and SNVs, and then maybe also identify if there is any mosaicism in some of the known genes. This has not been examined in this data set for congenital kidney defects at all. But in addition to this, as we're reviewing the data, maybe we should also look at some of the genes involved in other congenital defects to see where perhaps we can augment our diagnostic yield in this patient population. And then we hope to be able to do, discovery of new genes and syndromes by doing analysis of coding and non-coding de novo variants, and then replicate these signals in additional cohorts that we have available, either via looking for de novo variants or, again, looking for case control analysis, and hopefully collaborate with others in this group. So I look forward to getting feedback from everyone and look for opportunities to collaborate and learn from you all. And that's it.
Thank you, Dr. Gharavi. That was such a great way to kick off this webinar. Thank you so much. We're going to move forward to the next presentation. I encourage people to start asking questions of the presenters in the chat, but we'll have a question period after our next two speakers. So with that, let me introduce Dr. Xiang Wang, representing the structural birth defects and childhood cancer. So this is a cohort made up of children who have both a childhood cancer and a structural birth defect.
Thanks, Valerie. This is Patrick Sleiman. I'm going to do the first slide and then hand it off to Sharon. I don't actually have Wi-Fi here, so if we can go on to the first slide, the deck with the pie charts, that would be great. Okay, so guessing we're there, I'll start by saying that the composition of the CAD cohort is somewhat different to most others in this program, and that we're not focused on a specific phenotype. Our selection criteria here were to identify kids that had both the birth defect and the pediatric cancer, just as Valerie said, with the aim of really identifying shared autograph pathways. And so through the selection criteria, we ended up with a pretty sort of diverse group of cancers and birth defects that hit on every system, and those are plotted out on the slide that I'm hoping you can see. And so given the diversity of the phenotypes here, we've spent quite a bit of time and effort developing approaches to mine this data set. Today, Xiang is going to present two of those approaches. One is, and both are really central to the aims, but the first is to differentiate mutations that are causal of both, as opposed to a mutation that's more associated with a cancer or a birth defect. So that's the first challenge. And then the second is looking at a sort of more general phenotypic clustering approach that we're using to drive gene discovery really here by defining pseudo case control sets. So with that, I'll hand it over to Xiang, and he'll walk you through the rest of the slides.
All right, next slide, please. Hi, this is Sharon, a bioinformatician. Happy to have this chance today. As you know, our KIT first cohort has patients with cancer plus non-cancer phenotypes. And the natural simple question is, are there one causal variant or two variants? For a brief background, we have developed an in-house rare disease variant prioritization software called GD-Cross. In the past 3 years, it can efficiently rank the variants in the patient by their probabilities of being the causal variant. It is based on the HPO system and a bunch of other curated medical genomic resources. Now, specifically for our KIT first cohort, we expanded a part of the GD-Cross functions to quantify a variant's explanation powers for the two phenotype groups and analyze them in a 2D plot. For example, in this case, on the left side, we mapped all the HPO terms in HPO's hierarchical structure and grouped them into two families. The blue ones on the left to be non-cancer terms, and the red ones on the right to be cancer terms. Then in the patient, we consider a handful of candidate variants which have passed preliminary filters. Each of them was evaluated by how well it can explain the cancer than the non-cancer phenotypes. All of the quantified explanation powers were then normalized to a 0 to 1 scale. 0 means it cannot explain the phenotypes at all, and 1 means it's perfectly perfectly and very specifically explained all phenotypes. We plotted these normalized explanation powers in such a 2D square. Each dot is one variant. When more than one variant occupies the same position, the color gets darker. The x-axis is the explanation powers for non-cancer phenotypes, and the y-axis for cancer phenotypes. It's just the colors and the shapes reminded me of the beautiful cherry blossom in Washington, D.C., and there comes the name. This very case on the left side is one example of the scenario I call the scenario 1, when one variant explained both sides. Specifically, in this case, the answer is in WT1 gene. And here on the right half of the slide, don't forget that we can also have a scenario 2, when two variants explain each side separately and near independently. When this happens, this pattern or something similar will show up. You will see some variant sit very high on the y-axis and something else sit on the right end of the x-axis. In this case, a variant in RUNX1 explains leukemia and a variant in PKD1 explains birth defects. So these are the basics for the cherry blossom plot at the single case level. Next slide. We can certainly plot a cohort together. Here we picked 840 cases in our KF cohort, quantified their 9,000 prioritized variants, and plotted it in one graph. The top left is the one, and on top right, there's a small control cohort, which is basically childhood rare disease, but no cancers. In the distribution pattern, the two major differences were found. One is in the KF cohort, there are more variants enriched right on the y-axis, which means they explain cancers only. At the same time, we're more interested in there's a top right red box. We also see more variants in KF cohort enriched there, which means one variant can explain both sides. The control cohort used here is pretty small, and we're going to find some much larger ones to further confirm these findings. Now, on the bottom panel, you see something a little different. Here, we still plotted the whole cohort, but only to limit it to a chosen gene. This gene-specific plot could help us understand the gene behavior in the cohort level. The example I show here is how NF1 pathogenic variants behave in our cohort, in our KF cohort. Most of them, you can see on the y-axis, is pretty high. They have medium to high explanation powers for cancer. And the explanation powers for the birth defects scattered actually from very low to medium to very high. It depends on the specific phenotypes of a case. Next slide. Instead of working at the cohort level, we can also work at subcohorts. The basic idea here is to do phenotype clustering in a cohort to group similar cases together. As you have seen in a previous presentation, our CCBD cohorts is actually lack of internal similarity, but we can still find some patterns here. And then any further analysis, including the 2D cherry blossom plot, will have lower background noise in that subcohorts. On the left side, that big one is the hierarchical clustering of 1,464 KF programs by their assigned attribute terms. You can see clusters on the diagonal. On the right side is just a zoom in of one of the tight cluster with many solved cases and many unsolved cases. The solved cases can serve as anchors to the cluster. Assume a cluster is a real cluster, then the causal gene and even the shape of the 2D cherry blossom plot should be similar for most of the cluster members. And this information will be highly valuable to the efforts to solve difficult cases and discoveries for novel causal variants or genes. Next slide. To summarize, we're going to resolve individual cases with the guidance of the 2D cherry blossom plot and also the phenotype clustering. And we're going to study further genes which enriched in that 2D plot's top right corner. We will identify due his cases and novel birth defects of cancer predisposing pathways. And for new findings, we'll certainly do wet lab validations and functional studies. Under these two strategies, I'm quite sure there will still be a lot of unresolved cases. And for those cases, we will use AI and deep learning algorithm to follow on. Finally, this whole thing can be made a discovery machine, and we expect this machine to be portable and can be used in other cohorts. Thank you.
Wonderful. Thank you so much. You have a question in the chat, and we can address it in the question period too. But with that, we'll move on to Dr. Sharon Diskin, representing the neuroblastoma project.
Great, thank you. So thanks for inviting me to share some of our data and phenotyping efforts here today. What I'll be presenting is really a collaboration with John Maris at CHOP and Andy Olshan at UNC. So our project focuses on neuroblastoma, and this is a malignancy of the developing sympathetic nervous system, typically involving the neural crest. The median age of diagnosis is only 17 months, and it's actually the most common malignancy diagnosed in infants under 1 to 2 years of age. Only about 1 to 2% of cases report with a family history, and we consider these to be familial neuroblastoma, whereas the vast majority are thought to arise sporadically. Neuroblastoma has been observed and associated with other conditions, particularly those of neural crest-derived tissues, for example, Hirschsprung's disease. Neuroblastoma has also been associated with multiple congenital anomalies. And recently, we published an association of the 16p11.2 microdeletion that we also heard about in an earlier presentation with neuroblastoma. And this had a fairly high effect size. And it was really our first look at some of the kids' first data where we were able to show that these appeared to arise de novo when parental DNA was available. So neuroblastoma has a hallmark of clinical heterogeneity that's really thought to reflect the underlying genetic heterogeneity. So some neuroblastomas can undergo spontaneous regression. And then at the other extreme, kids diagnosed with high-risk neuroblastoma often have really aggressive refractory disease and ultimately succumb to that disease. And over the last 10 to 15 years, our sort of model of genetic predisposition has evolved. And we tend to think of it as this figure on the right, where we plot the allele frequency on the x-axis and an effect size on the y-axis, and the different mechanisms or approaches we've taken to identify predisposition variants and candidate genes, ranging from common variants at the lower right that were identified by genome-wide association study and implicate this wide array of genes in the bottom right, to the other extreme where studies of familial neuroblastoma initially led to the identification of FOX2B through comorbidity with Hirschsprung disease back in 2004. And then really the identification of the main neuroblastoma familial predisposition gene, ALK, through a linkage study. And then there are a bunch of genes and variants that have been identified through existing sequencing efforts, either exome or whole genome, of really unknown effect size right now. And one of the real limitations we've had and struggled with was, in terms of understanding sporadic neuroblastoma, was that we typically don't have parental DNA to assess heritability of these variants. Next slide, please. So this was really one of the main goals of our XO1 sequencing application. So through partnering with Andy Olshan at UNC, who led the neuroblastoma epidemiology in North America study, or NINA study, he had recontacted families of kids with neuroblastoma, had them complete detailed questionnaires, but importantly also collected saliva DNA both the affected child and the parents, if available. So through Kids First, we've sequenced through whole genome sequencing over 1,500 of these germline samples, representing a heavily QC'd set of 556 probands. 457 of them have both parents sequenced as well, and 99 have either just the mom or the dad sequenced with the proband. And then we're really also interested in understanding sort of host tumor interactions and understanding progression of neuroblastoma. So through the Children's Oncology Group Nucleic Acid Bank, we were able to obtain tumor DNA for 336 of these individuals, which underwent whole genome and exome sequencing. And in parallel, RNA was available for 207 tumors that underwent total RNA sequencing. So overall, there are 201 probands with full characterization in the Kids First cohort. Next slide. So this is a rather complicated slide, and I won't go over it in great detail, but this is sort of our initial analysis approach to this cohort, where on the left, our primary focus is to study the neuroblastoma triad and dyads. Here we're looking for both inherited and acquired de novo pathogenic variants, as well as evidence for mosaicism. Our plan is to look at both protein coding and non-coding regions of the genome. The non-coding aspect will be facilitated through integration with the data sort of illustrated in the middle, in particular what we refer to as the neuroblastoma ENCODE and some 3D chromatin data that we have. And then in parallel, we are studying the tumor DNA and RNA, both in terms of host tumor integration as well as just the tumor phenotypes. And our goal is to then integrate these with other childhood cancer data sets to identify pleiotropic risk. And ultimately, it would be great to integrate this further with structural birth defects through Kids First. Next slide. So in terms of the clinical and phenotype data we have, the majority of these patients were also enrolled in the neuroblastoma biology protocol through the Children's Oncology Group. Therefore, we have sort of standard clinical and biological characteristics, including the gender, the diagnostic category, the site of the tumor, age of diagnosis, the Children's Oncology Group risk group, staging for the tumor, histologic data, as well as some other tumor biologic data. And not listed here, we have patient survival data. And this has really been our primary focus so far in understanding some of the pathogenic variants we're seeing in the Kids First cohort. In parallel to that, we're working with Andy to integrate the data from his questionnaires in the NINA project. So here he's gone back and obtained sort of general background information on parents, detailed history of the pregnancy, the child's birth and early feeding, family history of cancer, including but not limited to neuroblastoma, tobacco exposure during pregnancy, vitamin usage, as well as foods and beverages ingested during pregnancy. So it's a really quite detailed questionnaire that we're working on structuring and integrating with the Kids First genomic data. Next slide. So I just wanted to share some unpublished data that we've looked at thus far and integrated with some of the COG clinical data. So Emily Blau is an instructor here at CHOP and Penn, and she sort of devised a set of 198 cancer predisposition genes, which Zalman Voxman, who's a bioinformatician here, has analyzed the Kids First data and assigned pathogenicity to each of the variants. And overall, we're seeing a little over 13% of the cohort harbors at least one pathogenic or likely pathogenic variant in a cancer predisposition gene. And then we were able to look really for the first time to see if these were inherited or acquired de novo. And the vast majority appear to be inherited without a statistically significant difference in paternal versus maternal inheritance. And only 3% appear to arise de novo, which is a relatively small number. Q advance, please. So our next question was whether the patients that harbored a pathogenic germline variant were sort of enriched in any of the clinical or biological characteristics defined by the Children's Oncology Group. Next slide. So what we found was that they indeed were, and we found that those patients with a high-risk neuroblastoma or an advanced stage tumor or MICA amplified tumor, which is a very potent oncogene in this disease, were more likely to harbor a pathogenic germline variant. So this is sort of our initial look and integration with some of the clinical data. And like I said, we're now looking to expand this beyond SNVs and indels to structural variants, and also incorporate the more qualitative data from the NINA study. Next slide. So in summary, we know neuroblastoma is a complex genetic disease, and the diverse clinical course very clearly reflects underlying genetic heterogeneity, both in terms of germline and somatic variation. We know that common variants identified by GWAS correlate with clinical risk groups and outcome in neuroblastoma. We also know that rare variants in the germline can either be acquired de novo, such as the 16p deletion, or they can be largely inherited, such as the rare SNVs and indels that we're seeing in the cohort. We also know that there are multiple comorbidities that have been observed with neuroblastoma, but we don't have extensive phenotype data on this cohort in order to study it further. So we've been thinking for several years now to propose a COG epidemiology study with a detailed questionnaire. And the thought would be that this would encompass the Kids First cohort, those patients included in the genome-wide association study, as well as the NCI target project for neuroblastoma. And we know for certain that all the Kids First families here have consented to be recontacted because by definition, Andy has already recontacted them all. And we know from past experience that a large subset, but not all, of the GWAS and target cohorts have also, families have also consented to recontact. So we estimate about 5,000, might be slightly lower. And our goal is to obtain a detailed family medical history and data collection, including neural crest-derived phenotypes, congenital anomalies, neurodevelopmental phenotypes such as autism spectrum disorder and others. And also data regarding treatment response and related comorbidities that I didn't really talk about here today, but is also of interest to us. And I'll just end by saying that I think additional phenotyping efforts could really be facilitated and enhanced through use of a COG approved online phenotyping system, and maybe Kids First is the way to sort of drive that forward. And this would allow for swift data entry and integration with other Kids First cohorts. Next slide. And I just want to end by acknowledging everyone who's contributed to this work so far, especially Emily Zalman, Laura, Alex, and Rebecca, who have done most of the data analysis and interpretation. And obviously our collaborators, John Maris and Andy Olshan, our funding sources, and the great group at the Kids First DRC. Thanks.
Thank you so much, Sharon. So we're in a question period right now. I think we'll give ourselves the 10 minutes if we want it for questions. Before we look at the chat, does anybody want to unmute and ask a question of any of these speakers?
This is Joe. I can ask my question. I posted it to the chat, but I can ask it to. This was a question for Abhi.
Yeah, go ahead.
Okay, I was wondering about your slide on the CNVs, and maybe it's kind of a general question for many of the groups, but many of those CNVs that you discussed were previously implicated in neurodevelopmental disease. So I'm wondering if you're planning to try to hone in on the gene in particular, or genes within the interval that are the risk factor for the kidney defect. Do you think that you're going to find second hits in the other copy of the gene on the other chromosome, or do you think there's going to be kind of trans modifiers? Is it too early to say? Thanks.
Thanks for this question. I think this is a really interesting question. I think, well, Simone Sanakirky in our group wound up doing this in terms of the fine mapping for the 16p11 and identified the mutations in TBX6 as one of the drivers for congenital defects within that locus. So I think that it certainly is going to be possible. We just need large sample sizes to be able to do the fine mapping, and then also do independent sequencing to find maybe intergenic mutations that will help us narrow down on specific genes. So we're certainly interested. I think this question of modifiers is also very interesting, but as you know, you need probably larger sample size, and this is the opportunity, I think, here in this and within this group to try to get to some of these questions now and identify what's going on in terms of this variation in phenotype.
And Drs. Wang and Sleiman, I wonder if you want to try to address this question Melissa raised about you used HPO structured data to do the analysis you showed, but have you used NCIT?
Yes, I saw the question. I'm not sure is Dr. Handel still here.
I'm here. I'm
okay.
here.
Okay. About the size of the terms we use for each case, I don't have the exact number handy, but it's about 1 to several. And we have never used NCIT because we have been based our system, our GD-Cross program on the HPO system. I don't know if NCIT also have the kind of term to gene associations information. If that is available, then that is also okay. But
so that's a really interesting question. So there is some encoding of gene information in the NCIT directly. So the structure is not exactly the same as what we have for the HPO annotations. But this is one of the challenges that we're trying to work out with NCI is how to best support a semantic framework that is inclusive of the sort of phenotypic features that we describe in HPO versus those that are in the NCIT, and really helping the two kind of co-evolve into being a better framework. So perhaps this conversation can be taken offline, but we'd love to experiment with you on some of these issues and come up with the best possible design. NCIT is a much richer terminology for cancer, and we really try not to put too much of the cancer work in the HPO for that reason, but for you with regard to the annotations.
Good to know, but I also trying to explain that we're not too worried about the number of HPO cancer terms, because in our software there's already a kind of balancing and normalization inside, so more terms is not a big problem.
Okay, terrific.
Hi, Valerie, this is Philip Lupo. I had a question for Dr. Diskin, or actually a couple of questions, if she's still with us.
I'm still here.
Great. I really enjoyed your presentation, and I had a couple of questions. The first was I was interested in the proportion of the patients that had a pathogenic or likely pathogenic variant in a cancer predisposition gene. I know that these estimates are turning out to be variable by tumor type. I was curious, though, about the list of cancer genes that you included. I didn't get to see fully on the slide. Was that a selected list specific to neuroblastoma, or was it a larger list that included genes that people have or haven't thought of as being related to neuroblastoma?
So it was a relatively inclusive set of 100. The only genes that were displayed on the slide were the ones that had a P or LP variant. But the larger set of genes was a set that Emily curated, not necessarily directly known to be involved in neuroblastoma, certainly. So it's a little more inclusive than maybe some of the other published papers that look at pathogenic germline variants in cancer predisposition genes, but they all had evidence of being linked to cancer predisposition. They also included the genes identified through genome-wide association study in neuroblastoma, so that was also another subset of them.
Very cool. The next question I had was more about your future plans, and I hope I get to pick your brain and help facilitate that as you know, being involved in COG. But with your plans to reach back and get additional phenotypic information on the cases that were part of NINA, did I misunderstand that, or did Andy, as part of his study, collect information on some of these co-occurring phenotypes like congenital anomalies or autism spectrum disorders? Or
No, unfortunately, he collected a lot of data, but his interest at the time was more in sort of the folate pathway and influences during pregnancy. And actually, I've been talking with him for several years about recontacting, families to look in particular at neurodevelopmental disorders and congenital anomalies. And I think we've submitted a couple proposals that haven't been funded, and I think now we've finally gathered enough data and evidence to really justify it much better. So we don't have any data on that right now. So that would really be a goal of going back and recontacting the families. And our thought in the past was to include everyone in the GWAS as well as the target project. Because these are rare and it's just hard to get the numbers, especially in a relatively rare cancer.
Yeah, I would suggest that maybe you have this as part of your strategy, and I'm happy to talk to you more about this offline. But of course, these individuals wouldn't have existing sequencing data, but all of the kids with neuroblastoma that are getting consented to Project Every Child that could be contacted and enrolled in a study to get medical records and administer questionnaires, and you'd have the banked biological specimens, at least on a subset, that would be collected as part of Project Every Child. If neuroblastoma is part of Project Every Child, I think that it is. But anyway, we can talk more about it. Yeah.
Yeah, thank you. That would be great.
Of course. Thanks.
Okay, it's 4:05. Unless there's another burning question, I think we should take our break. Is it okay with folks if we actually turn this into a 5-minute break and come back at 4:10?
Yeah, that's fine.
That's fine.
Okay, let's do it. So we'll come back at 4:10. I'm going to make a pun. I'm so sorry. We're going to charge ahead with the TARD syndrome presentation at 4:10, and we'll see you guys there.
Thanks, Valerie.
Thank you to our first round of speakers. This was great.
Well, Valerie, I was saying I'm also online if there are any questions. This is Hakon.
Oh, hi, Hakon. Perfect. That's great. Yeah, I think the question Melissa raised was really interesting, and I wonder if let me touch Allison, but yeah.
But I mean, the HPO terms, I mean, it's actually interesting that GD cross, the more HPO terms we have and the more granularity we have on phenotypes, the better the program performs in terms of accuracy and in terms of, priority or ranking variants. But I mean, using that other sort of more cancer phenotype, cancer-related system may be actually beneficial for this project.
Yeah,
oh, go ahead.
This is Melissa. Hi, I'm still here. I think that would be really fantastic to try to do some experiments to help facilitate that. We, the structure of the NCIT is different, and so, I don't think there's been a lot of experimentation about using the same kind of semantic similarity or enrichment kinds of techniques using the NCIT, but the sort of descriptions within the NCIT are much richer description in terms of the semantic structure for cancer. So we probably need to do a little bit of innovation on the algorithm. I think the combination of the two would be really quite impactful. There's some really bad background. Sorry about that.
Yeah, no, the program is currently sort of tailored towards the HPO, but I mean, it wouldn't take much to tweak it for the.
Yeah, I mean, I think it's really interesting to think about how do you do it across kind of the phenotypic like methods, and then the cancer community also has methods where not necessarily NCITs tying to the genes, but you have all the like civic and cosmic and other resources that are tying the different types of cancer diagnoses to the different causal genes, and how do you really do that, combining the two? I think it's very interesting and kind of a novel thought. I think that's really happening here.
Yeah.
Melissa, we were talking today with Justin Reese, and I actually was showing him some of my slides from earlier in the presentation on the summary of the phenotype similarities, and he's also interested in trying to see if knowledge graphs can help with these classifications, of course, but
Fantastic. Can we just make a little action item to maybe have a small group follow up on this topic and kind of design some next steps and maybe a small experiment to kind of get us going? Because I'd love to use this as an opportunity to help inform how we kind of create the framework longer term. We've been dabbling around the edges of how to best do this for a while, but we don't actually have enough sort of requirements to make sure that the current thinking is actually really fit for purpose. So you guys could potentially be our best guinea pigs if you're interested.
Yeah, absolutely. I mean, I think that would be great.
Terrific. So
Allison, I wonder. Oh, go ahead. Sorry.
No, you go ahead.
I was just going to say, later, Allison is going to. Sorry, just really quick, she's going to demo the portal a little bit to inform some of these discussions, and maybe she can just show us some of the differences of using an NCIT-based search versus an HPO-based search later.
Yeah, that would be really helpful, actually. I think that would spark some ideas for future integration work. Yeah, absolutely.
I just want to thank Melissa for the NCIT thing. I'll take time to read it and to see if I can bring some fresh air from it.
And we all padded our way through the break. It's 4:10 already. So assuming everyone's back, let's go ahead and get started. Nicole, can you advance to the next slide? Let me introduce Dr. Donna Martin, who's the PI of the TARD syndrome cohort, which is a fiscal year 19 cohort. So it's a newer project.
Great, thank you. Hi, everybody. So I'll be presenting our project. Also on the call is Dr. Stephanie Bielas, who's an associate professor in the Department of Human Genetics, and Jacob Ogle is our research associate who's been doing the heavy lifting on this project. I'm affiliated with the Department of Pediatrics, currently the chair, and also affiliated with the Department of Human Genetics. And we've had a longstanding interest in CHARGE syndrome. Go to the next slide, please. Oh, let me go to full screen. I don't know quite what happened. CHARGE is a rare genetic disorder. It's a multiple anomaly condition, and it's defined by the acronym CHARGE, which stands for coloboma of the eye, which typically affects the retina, but it can also affect the iris. Heart defects, which vary from tetralogy of Fallot to ventricular septal defect, double outlet right ventricle, and a variety of other types of congenital heart defects. Choanal atresia, which leads to breathing difficulties in newborns. And then the R is for retardation of growth and development. The G stands for genitourinary malformations. And this commonly presents as genital hypoplasia, later with delayed onset of puberty. The ear is for ear abnormalities and hearing loss, which typically manifest as a combination of sensory neural and conductive hearing loss, and is also associated with external ear malformations. In addition, these children have craniofacial malformations, and collectively this condition affects about 1 in 10,000 individuals worldwide. Back in 2004, the gene CHD7 was identified as causative for CHARGE syndrome. CHD7 is a chromodomain helicase DNA binding protein. However, since then, we've learned that somewhere between 10 and 30% of individuals with a clinical diagnosis of CHARGE syndrome do not have an identified pathogenic variant in CHD7, which really prompted the question of whether another gene was involved or other genes, or if this was associated with some other underlying cause. Next slide, please. So our goal for this project was to see if we could identify new genetic causes of CHARGE syndrome, and also to expand our understanding about the phenotypic expression of CHARGE. Over the years, people have asked whether the identification of a pathogenic variant in CHD7 is necessitated by having all the features of CHARGE syndrome, a subset of CHARGE features, or whether you could even have isolated features of CHARGE syndrome and have a pathogenic variant in CHD7. And what we have learned is that there is probably a much broader phenotypic expression of CHARGE syndrome than was previously recognized, but that is one of the underlying goals of this project. Next slide, please. So our research pipeline includes a variety of patient sources. I'm a clinical geneticist, and over the years have collected many of these children through my own clinical work at Michigan Medicine. I'm also active in the CHARGE Syndrome Foundation, which is an international organization that provides support for families with CHARGE syndrome, as well as professionals who take care of these children. And we have collected saliva samples through our interactions with individuals at the family meetings for the foundation. In addition, over the years, we have collected samples through collaboration with other individuals and have been using those samples that we've collected, as well as recruiting individuals going forward. Our sequencing sources for this previously were collaborations with the University of Washington. We had pursued some whole exome sequencing, and those data are just now under analysis. But through this, we developed a cohort of individuals who had clinical features of CHARGE syndrome or subsets of CHARGE features, but were actually whole exome negative. So the idea here was to take those individuals and pursue whole genome sequencing through the Gabriella Miller Kids First project. Next page, please. Next slide. Our cohort is as follows. We have blood-derived DNA from about 148 families, and saliva-derived DNA from another 17 families. So some of these come in as trios, some as duos, and some as singletons. But this is the cohort that we're looking to get sequenced, and then ultimately take those sequence data, try to identify pathogenic variants or likely pathogenic variants in novel genes. We suspect we may uncover some CHD7 variants in genes, even though our cohort is enriched for individuals who previously tested negative for CHD7. We've already had the experience of identifying pathogenic variants in CHD7 that were actually missed by previous sequencing. Sometimes that was done on a clinical basis. And then we also are looking at the possibility for CNVs that will come out of these data. Go ahead, next slide, please. So our phenotypes are derived from multiple sources. We have access to the medical record from Michigan Medicine patients, and also access to clinical data that our collaborators provide for us. Our phenotypic questionnaire is stored and transferred into a REDCap database, and those data can then be mined or queried by research personnel with appropriate access to the medical record. And we're really looking forward to see how broad these phenotypes are, the ones that are associated with pathogenic variants in CHD7 or other genes, and then collaborating with other groups to see if we can harmonize data across the various Kids First data sets. Next slide, please. I think that was it. Okay. Happy to take questions. Now we're at the break.
Thank you. We'll go ahead and blaze ahead. So our next speaker is Dr. Stephanie Ware, speaking on this laterality birth defects cohort.
Well, thank you for asking me to speak, and you can go ahead to the next slide. So laterality birth defects are a very interesting arrangement of congenital anomalies that are caused by an early developmental disorder. So very early in embryonic development, before the organs are even formed, the embryonic axes are established, and the left-right axis is the last of the three body axes to be established. And if the left-right axis establishment doesn't go correctly, then the placement and formation of the organs of the thorax and the abdomen occur inappropriately. And so really clues of disorders of laterality are complex heart malformations, because the heart is particularly susceptible to abnormalities of left-right patterning. And then abnormalities of the positioning of a variety of other internal organs. So what you can see on this slide is the normal arrangement on the top left, the normal arrangement of the thoracic and abdominal organs, with the lung having 3 lobes on the right and 2 lobes on the left. And when you have mirror image arrangement of the organs, that's called cytosine versus totalis. And then really all of the other combinations, which are kind of ambiguous arrangement of the organs with respect to the left-right axis, would be considered heterotaxy. And interestingly, individuals with heterotaxy, also about 40% of them will have other type of birth defects that fall in the midline, and those may include rib fusions, vertebral defects, and also in rare cases, neural tube defects. And we know from some of our animal models of heterotaxy that exencephaly and other neural tube defects are more common in those model organisms than what we see in the patient population. And so the genetics of heterotaxy has been fairly well studied from an animal model standpoint, and many developmental pathways have been identified based on that. So I'm going to talk really much about the genetics today, but we and others have published on Mendelian mechanisms, including CMVs and genomic disorders that are associated with heterotaxy in somewhere between 10 and 20% of patients, X-linked inheritance that occurs in about 5% of males with heterotaxy, and then autosomal recessive and very rarely autosomal dominant with reduced penetrance. But far and away, the most common inheritance pattern seems to be complex or multifactorial inheritance, with heterotaxy thought to be almost a purely genetic phenomenon with small environmental effects with maternal diabetes and disruption of vasculature. So the goals of our study are really to perform genomic analyses in 550 probands with a spectrum of laterality defects. And embedded within that cohort is 105 trios. And a subset of these have had exome and are exome negative. And then one of the things that we want to do in terms of looking at oligogenic or more complex inheritance is really to look at some of the known developmental pathways and candidate discovery genes to look at burden analysis, knowing that the mechanisms of heterotaxy include things like ciliary abnormalities, cell polarity defects, and abnormalities of TGF-beta signaling pathway. So this is just a screenshot of our REDCap database that shows some of the data set items that we're collecting. We do have a pedigree that's done on all of the enrollees and questionnaire data associated with that related to family history. A lot of phenotype data collected because of the multiple abnormalities that can occur in these patients. And then deep phenotyping of the cardiac malformations along with any clinical genetic testing that has been performed. So these are some of the questions that we ask in obtaining the pedigree and going through the questionnaire with the families. Most of these are geared around trying to identify any other type of birth defects within the family tree that might be associated with laterality defects. And we actually do see an enrichment of other types of birth defects in these pedigrees overall, although it's very rare, as I mentioned before, that we'll identify a Mendelian inheritance pattern. So obviously we want to know detailed cardiac history for all family members. The pulmonary history is important, not because of the lung lobation issues, but because of ciliary abnormalities that are associated with a subset of heterotaxy cases. And actually patients with a diagnosis of primary ciliary dyskinesia, although if many of them have laterality defects, it'll be situs inversus totalis, there are a portion of them that have heterotaxy as a manifestation. So questions about chronic wet cough, chronic ear infections, sinusitis are all things that go along with a diagnosis of primary ciliary dyskinesia. Other neonatal conditions, one of the common things that you see in patients with heterotaxy is biliary atresia or abnormalities of the biliary tree, and as well as infections that can accompany abnormalities of spleen function. So that's also some of the questions then we ask about chronic medical conditions. So this is just an example of a typical 3-generation pedigree that we obtain, although this is actually an atypical pedigree because, as I've said a few times now, it's rare that we get familial cases. But you'll note in this pedigree, the 2nd generation, the parents there is consanguinity in this pedigree, raising the higher likelihood of autosomal recessive. And so this is an example of a pedigree where there's heterotaxy. In one of the individuals, there's a child with bilateral ear tubes, which of course is common in pediatric patients, but does raise some potential suspicion for a a ciliary abnormality, and then another child that had left isomerism. And then there's more distant relatives with situs inversus. So with regard to the cohort in a little more details, we've broken the cohort down. What's shown at the top left there is the trios and then the singleton cases. And basically we have cases that are what I would call classic heterotaxy, which means that they have a heart defect consistent with heterotaxy, as well as evidence of abnormalities in other thoracic or abdominal organs in terms of positioning or function. And then we have a smaller portion of the cohort that has just isolated congenital heart defects that fall within the heterotaxy spectrum. And there isn't a pathognomonic heart defect that for heterotaxy, but there's a range of heart defects that are consistent with the heterotaxy spectrum. And then we have embedded within this cohort a much smaller number of patients that fit clinical criteria for Bacterial, which many of you know is just an acronym that describes the congenital anomalies that occur. But these patients both fit criteria for Bacterial as well as criteria for heterotaxy. So that's the smallest subset of the cohort. And then the classic heterotaxy patients, and then finally those that have just congenital heart defects. And listed on the right are the types of heart defects that fall within the laterality spectrum. So I would say some of the challenges that we've had in terms of the integration of the data and harmonization, I'll go through here. And on the right, what you see is a count of sort of the different types of abnormalities that we're seeing that we have collected in our cohort. And most of the individuals in the cohort have multiple abnormalities, as you can imagine. So for example, when we have skeletal abnormalities, vertebral abnormalities listed as 34 individuals in the cohort, all 34 of those individuals will also have heart defects and other evidence of laterality defects. So the phenotypic heterogeneity, I think, is a real challenge in terms of classification. And I was really interested in the first talk from today about what the Kids First cohort is doing. And I'm really interested to see how the HPO terms from this cohort are going to map with other collected cohorts. And that was actually one of the things in submitting our samples for this study. We were hoping that we would be able to identify both phenotypic and genetic subsets of this cohort that overlap with other congenital anomalies that have been collected in a more dedicated way. I think it'll be fascinating to see if there's a genetic overlap in addition to the phenotypic overlap. The fact that this is a rare disease, so heterotaxy is thought to be about 1 in 10,000 and situs inversus totalis about 1 in 20,000. So we have about between 10 and 15 cases at our medical center every year. So much of our recruitment is from external sites. And while we do review all of the imaging information that we can get, there's just lack of standardization, as you can imagine, that makes things a little more challenging. There's very confusing nomenclature. There's multiple defects or arrangement of organs called by the same name. So people often are using the same terms to mean different things. And even though that first slide that I showed made it seem like there were clear buckets or bins that the organ arrangements occur within, and in actuality, almost every individual is unique with regard to their specific anatomic arrangement. Historically, then, there was a lack of ICD codes to cover heterotaxy. Didn't used to be a billable code. Now it is, but whether people use that code or use the subset codes or the specific congenital heart defect code, for example, is variable. So that makes EMR searches problematic. And you have to do, if you're trying to identify eligible patients, you have to sort of combine your searches to ask for multiple abnormalities. And finally, the cardiac phenotyping. I'm not going to go through this in detail because as I was reviewing the last webinar, Betsy Goldman's did a really terrific job of describing cardiac phenotyping that they've done for the PCGC cohort. And just the idea that there are very many different classification systems for the wide spectrum of cardiac malformations, and they have each has different strengths and weaknesses, but they vary. And so the PCGC has used Filer codes. We've been using BOTO codes, which are codes that are used by the National Birth Defect Prevention study. And so that's going to make some of the harmonization of the data a little bit difficult, I think. And I think the cardiac phenotyping really requires high-level expertise. There's just many of these really require a pediatric cardiologist, despite the number of SOPs you put in place to try to make rules for these. But next slide or button. I think the real opportunity here, sorry, I had one more thought on that previous slide, if you can go back, in the blue text up below. I think the real opportunity here is for identifying phenotypic and genetic subsets, as I already mentioned, for multiple anomalies. And with that, I'll end. The last slide is just acknowledging the people who have contributed importantly to this. Thank you.
Thank you so much. I definitely have thoughts and questions, but let's keep blazing ahead to our third talk for this session. Dr. Joseph Gleeson, who has two cohorts, one that's focused on structural brain defects and one that's focused on neural tube defects. And then we'll have a question period after this talk.
Great, thanks so much. Thanks for the opportunity to present. And I'm really excited to be part of this group and to share experiences. My training is in child neurology, and we've been ascertaining families with a range of neurodevelopmental disease for many years. We put two cohorts together for Gabriella Miller, one that's on structural brain defects that's primarily recessive, because we have a large collection of patients that have been through exome sequencing that are negative. And the second is in a new cohort that we constructed recently that I'll talk about on neural tube defects. Okay, next slide, please. So first I'm going to present is on whole genome sequencing in neural tube defects. So neural tube defects are the most common CNS birth defect. They're present in around 3 in 10,000. It used to be more like 5 to 6 per 10,000, but since folate was introduced into the diet in 1998 in the US, the rate has actually dropped by about half. That's pretty impressive. Clinically, you can see these open neural tube defects at birth. Actually, they're seen quite often during fetal ultrasound. And it's generally considered to be a complex disease with not very much substantial work published so far into causes. We decided to try to tackle this through a specific hypothesis that de novo loss of function mutations contribute to risk, because the disease had a lot of the hallmarks of other de novo diseases like autism, congenital heart disease, and other kinds of structural disorders, and that the mutations are probably under strong purifying selection. And so we put together specifically a trio cohort that consists of a father, mother, and child. And the child with spina bifida must have the open neural tube at birth with the presence of hydrocephalus. And actually, most children with meningomyelocele have hydrocephalus at birth. Ours requires that a BP shunt is required to treat the hydrocephalus. So we were allotted about 1,000 genomes this year for roughly 333 trios. Most of these families have not had any genetic evaluation so far. The criteria for inclusion are that both biological parents must be available for dealing with either saliva or blood. Primarily, we have blood samples. There's maybe 10% of the cohort that's sampled with saliva. And I already mentioned the diagnostic criteria. And about half of the subjects that we recruited were conceived before and half after folate was introduced into the US in the grains. And so this actually makes for a nice hypothesis that we can test, and that's the mutations might be different in the trios ascertained before and after folate, because we see the risk has substantially dropped. So what has that done to the mutations? And that was a major emphasis of our proposal. Next slide. Can you go to the next slide, please? Oh, this is the next slide. Sorry, go back one. So the phenotype that we have is pretty light, actually, because we have very strict entry criteria, we want to take all comers. And the phenotypes, the information that we have from the historic part of the cohort is pretty minimal because those were all collected prior to 1998. And it's difficult to go back and re-phenotype those. For individual families, it's quite straightforward to do it. But all the families we've been ascertaining prospectively, we have all this information. We did a search in the medical records at Rady Children's Hospital here in San Diego and recruited, I think, around 300 trios using ICD codes. And we have all this information, including level of spinal lesion, any medication, seizure history, health status, etc. We're trying to collect maternal prenatal folic acid status. Well, that's published and already known to be very difficult to be trustworthy. Because by the time the mother knows she's pregnant already, the neural tube is closed in the vast majority of pregnancies. So we're also collecting family history and including other history of birth defects. Next slide. The second cohort that we're using is in recessive structural brain defects. So we've been studying structural brain defects for a long time. These are disorders where the brain is not morphologically normal, and generally that's appreciated at MRI scan or CT scan of the brain. These are not overt malformations like encephaloceles or something like this. I'd say microcephaly makes up a large percent of the cohort, but there's also migrational problems in the brain. These include forebrain, midbrain, and hindbrain abnormalities that are detected at the level of the MRI scan. And the goal of this project is to identify new causes. Most of these families have been through exome sequencing and they were negative. And we know that there's tremendous number of genes that can underlie this class of disease, probably thousands. At this point, we're well over the hundreds. And it can be very difficult to assemble uniform cohorts because the phenotypes are so poorly defined at the level of the MRI scans. So we oftentimes use genetics. We most often use genetics to help sort this out. You can find families that have the same gene mutated and then go back and find the similarities. So our inclusion criteria for this study are consanguineous. We're dealing both with multiplex and simplex families. Almost all these families are recruited from are abroad. And we have one or more affected children with a diagnosis of a structural brain defect and the presence of neurodevelopmental delay. We've been 1,100 genomes for this for 275 quads. We're trying to stick with quads as much as possible. This is father, mother, and 2 children, because as I've mentioned, we've already done the singleton exomes and they're negative. So we want to be able to find other rare causes that might elude exomes, and the genomes would be particularly helpful, for instance, structural defects, or even the occasional de novo mutations that are going to creep in. And we're hoping that we can find new genotype-phenotype correlations. Next slide. So the phenotypic data from the brain defects, we have a detailed intake form that we have on all the families that includes the entire pedigree, history of consanguinity. We have MRI scans on the vast majority of these, but they're in the form of JPEGs. They're not in DICOMs, which are the way that clinicians share scans. And that's because for historic regions in the Middle East, most of the families actually keep control of their scans, and they'll bring the scans in with them during the clinical visit, and then we can take pictures of the images that way. But that's the one limitation of the imaging that we have. So we have really good access to MRI data, but it's not the best quality images. We have medication, seizure history, EEGs, et cetera, other histories and developmental milestones, and then family history. Next slide. And I just put this at the end because this was one of the questions that we were kind of puzzling over. What kinds of tools would facilitate good genotype-phenotype correlations in our cohort? One of the things that we've been doing, and I'm interested to know if other people would share this idea, we have a lot of sequencing we've done already with genomes and some exomes, and we would like to know if it's possible to import that for a joint analysis rather than regenerate the sequence for both of our studies. And We have a lot of, as I mentioned, clinical intake sheets that are mostly on paper. So we'd like to know if it's possible to use some sort of software maybe to take excerpts from them that could be used in HPO terms. That would actually help a lot. We don't have a lot of review of systems. We have some really important kind of pertinent issues but we don't take a detailed family history, for instance, if it's not really relevant to neurodevelopmental disease. And we're also wondering if it'd be possible to build an RNA-seq or multi-omics workflow for joint data analysis or visualization, especially as our genome experience increases over time. That's the end of my talk, and I'll be happy to participate in the question part.
Thank you. There's a couple comments in the chat. People should feel free to unmute themselves and ask questions.
Joe, this is Donna Martin. I have a question. In your collection of patients, how many families do you have where there are 2 affected children? Are the majority singletons with an unaffected sibling, or do you have enrichment where more than one child is affected?
So for the meningomyelocele cohort, almost none of those are recurrent. The recurrence from meningomyelocele is maybe double the baseline rate, but it's not very high. So we don't have recurrences really in that cohort. In the recessive cohort, almost all these families are multiplex. It's rare that we'll recruit a family that's simplex. And in the Middle East, most families have 2 or more affecteds that we recruit. There are, of course, a lot of families with 1 affected, but our clinician colleagues don't bring those to our attention until we see a recurrence in the family. And that's because that really helps us with our solve rate.
Joe, I also have a question for you. This is Stephanie Bielas. Of the families that you have for whole genome sequencing for the structural brain abnormalities, how many of those do you have linkage data on that may allow you to hone in on genomic intervals to look for variants of interest?
That's a very good question, Stephanie. And I should acknowledge Stephanie because she really helped us build this cohort when she was in San Diego. So she's very familiar with this cohort. I would say we do linkage on the majority of the families, especially if there are, well, we don't want to have redundant data. So if we're going to do a genome on a quad and the quad consists of father, mother, and 2 affected, there's no reason to do linkage on them. But if there's additional unaffected members, then we will do linkage because that's an additional informative meiosis.
But if you potentially, if you had, you have DNA on so many generations that if you paired the linkage analysis, which is so many different meioses that allowed you to hone in on a single identity by descent interval, given the difficulty kind of doing whole genome analysis to find pathogenic variants, that might be an additional tool you could use that's unique to other data sets.
Definitely. And we have a project specifically on this, which is a multi-omics approach where we combine linkage, genome, and RNA-seq. And we see that can solve about half of our unsolved cases. So linkage is a really important part of what we're doing.
I'm just going to comment, Joe, that I really liked your idea about doing some joint analysis, leveraging existing data sets. And that's certainly some of the types of projects that with the NIH we've been supporting doing, and I think it's a great idea. So we can follow up and discuss what you think might be the best way to approach that.
Happy to do that anytime, and maybe other people have ideas about how that could be done.
Right.
Because what I'm really struck with in hearing the other talks is how similar in themes, even though we're all studying different diseases, how similar we're likely to find, common mechanisms, common threads, and common solutions that need to be applied.
Completely agree, and I think that emphasis on common is really part of the whole underpinning of Kids First, the hypothesis itself. So I think you're absolutely right.
This is Melissa. I just put a note in the text chat as well regarding the conversion of notes and text into annotations. We do have some tools and services, and we'd be happy to meet with you and help you come up with a good strategy for that.
And this is Valerie, getting back to the charge and laterality talks. You guys talked quite a bit about heart defects, and I'm glad you got to listen to Betsy Goldman's talk last time. One theme that came out of that meeting was maybe there's an opportunity for anybody who has heart defects represented in their cohort to work with Melissa Hendel's team and Nicole to do some sort of really in-depth alignment in Mondo. I should probably stop talking because I actually don't understand this, how this will work. Maybe Nicole can inform, but it is something that emerged from the last webinar.
Yeah, we'd be happy to work with anybody to make sure that we have all the diseases and phenotypes represented in Mondo or in the HPO as well.
Yeah, I love the idea of taking kind of the cluster analysis that Deanne did and using that to target different exo-1 groups for particular phenotypic areas to make sure that the modeling in both Mondo and HPO, as well as NCIT if it's needed, really kind of meet your needs, but do so in a way that's consistent across the different cohorts.
Yeah, this is Donna. I would echo that. Having the strength across projects is really exciting. I know for the cohort of individuals with CHARGE syndrome, we have overlaps with the clefting group, also with the structural brain malformations, because many of these individuals have cerebellar hypoplasia or corpus callosum abnormalities or forebrain anomalies, specifically in the olfactory bulb. So we're really looking forward to interfacing with other groups that have individual birth defects that have a lot of overlap with what we're looking at.
I agree with that as well. I think it's the same for the laterality cohort, and it'll be very interesting to do those types of analyses.
Deanne, did you want to elaborate on your comment about the RNA-seq analysis?
I just wanted to say that if anyone, there's several of us at Kids First DRC that can help advise on, how to, integrate data, do some RNA-seq analysis. I think kind of following up with what Adam said. There's several of us who can probably help. And if you want to contact me or someone else at Kids First, that's great.
And Dr. Gleeson, you mentioned you have these sort of crude scanned-in MRI images, but Dr. Martin, do you have MRI scans? I'm sorry, you told me at some point, and I can't remember.
I think on some of the patients we do have MRI scans. I'd have to get back to you on the specifics of that, but I know many of these children have had MRIs. I just don't know the numbers off the top of my head.
If this were volleyball, this is me setting up Adam Resnick for his closing comments later. But he knows that. Any other questions for this round of speakers? Okay, I think let's take our break, and it's 4:48, so we are ahead of schedule, which is really strange. But let's plan to come back at 4:58, and we'll start our brainstorming discussion then. Awesome. This is the brainstorming exercise. So I gave you the link in the chat, but Nicole's also going to share her screen. So you'll want to open the link though, so that you can contribute input.
Yeah, so if everyone can go to this link, it's linked from the slides here, the bit.ly/KF-discussion. And then when you get here, if you could change the layout, so right now it's vertical layout. If you click it to the horizontal layout, it might be a little bit easier to read and follow along. And luckily they have all this nice color coding on the left-hand side, so we can help keep track. So let's start. Are we ready to get started?
Yeah, I think so.
Cool. So let's start with the first question with the green solid box beside it. So, if everyone's able to get into this to the screen, does anyone have any problems? Let me make sure.
The color coding didn't show up for me, but it's okay. Oh. I'm okay with it.
That's too bad.
I can see it on your screen. Yeah.
Okay. Yeah. The first question. So what other data sets do you have or plan to intersect with your study? In other words, what other conditions will you study? Okay, we're getting some answers already.
So is this like a kind of a format where we can actually type stuff in here like a Google Doc? I haven't used this Bitly before.
Yeah, so are you on the Fun Retro site?
Yeah.
Yeah, so I think you should just be able to click the plus button and then add in any comments.
And if you need help with this question, I sent you a cheat sheet, a full list of all the Kids First cohorts in the chat. You know, obviously you didn't hear from all of them today, but I think it's cool that some of you have already inspired each other.
I want to just mention one thing. I can type it in here, but you know, RNA-seq I think would be a great way for us to interact, especially if it's done on similar cell types, because then we could each use each other's data set as a control data set if we could agree upon the cell type and the read depth and the methods that are used. I think part of what's so powerful about RNA-seq is if you have a large control cohort, then the differences in your individual patients can really stand out. At least that's our experience.
Yeah, great. Looks like people are jumping ahead to the questions. Does anyone have anything else about other data sets that you have or you plan to intersect within your study? We have RNA-seq mentioned by Joe, intracranial germ cell tumors, congenital heart disease, structural brain malformations, clefting, diaphragmatic hernias.
You know, a couple of other thoughts, you know, in that context, and I don't know if it'll spark anything, but you know, are there sort of thoughts around differences between sort of syndromic or chromosomal based data sets versus non-chromosomal data sets, or are there control data sets, GTEx, you know, sort of talk about RNA-seq or other types of data sets that people think are useful or routinely use or would like to use in the context of the comparisons?
Yeah, okay, some great answers here. What does NTD stand for?
Generally stands
for neural tube
defects.
Neural tube defects. Okay. It looks like you can vote too. So if you agree with some of these things that people are putting in here, you can add a +1.
The other thing I think could be really powerful, I'm hearing a lot of people with structural variants that are cutting across phenotypes, which is really interesting. 16, 21, other ones seem to increase the risk of a range of diseases, but we don't know how. And what would be really nice is if we could use each other's genomes as like let's say one study, let's say the kidney study finds 16p11 deletions, increased risk, and then my study also finds it in neural tube defects. Well, there's got to be something else contributing to it, and it'd be great to use each other's studies as using the 16p deleted patients as controls for each other's studies to try to find either cis or trans genes involved. Does that make sense?
Yeah,
I'm not going to type that in there, but
yeah.
That completely makes sense. I think that's how we can also identify modifiers, as you had suggested, using even looking at common variants, because that's one of the things that has been shown to exist even for congenital defects.
And I don't know if Sharon Diskin is still on, and whether from the cancer side that could also be something to think about, these chromosomal changes.
Yeah, Adam. Yeah, I'm still here. Actually, yeah, I totally agree, and I think it's a great idea for 16p and other regions probably. For us, they're very rare events, so it's really hard to sort of pinpoint anything. But yeah, I think it would be great to compare across phenotypes.
so in that light, is the methodology for calling SVs going to be made uniform between the different sequencing centers?
Probably not.
I don't know if there's uniform. I mean, they each use relatively standard pipelines to give you your basic VCF, but actually we're starting now to dig deeper into structural variation pipelines, and I think that, you know, based on the DRC's assessment, there's nothing uniform about it at all, but there's really an opportunity for a bunch of us to get together and discuss the pipelines people are planning to use and start sort of testing that against some of these data sets. You know, Dr. Gleeson, you are doing some long read sequencing as part of your project, but we're about to announce additional X01 projects that will be doing some long read sequencing, and we're definitely going to tackle this topic in that context as well.
Yeah, but this could be a great use case, you know, and that's really one of the roles and opportunities we have within the DRC is to help coordinate, you know, almost like a working group type of activity around these workflows that don't yet have sort of uniformity of consensus or functional equivalency. With a direct scientific output, like you just suggested, I think that makes it even more salient. And you're not the first one to ask this question, so, you know, I think it's, you know, to Valerie's point, a great opportunity for us to just take this on and do it.
As a point of reference, Harrison Brand will be presenting some structural variant analyses of the cleft X01 data at the online American Society of Human Genetics meetings.
Yeah, you guys got the plenary, or he got the plenary discussion. We're very excited about that.
Yeah.
Harrison also spoke at our spring webinar, and he spoke a bit about the structural variation analysis they did, and then Owen Hershey from Sharon Pond's group spoke a little bit about their structural variation plans as well. So I'll plug in the link to that webinar in the chat for you guys too, if you missed the spring webinar.
Good.
It's a good point.
We have to acknowledge that the methodology for structural variant calling is, shall we say, still evolving.
Right, and you'll see that the theme in the previous webinar was really using multiple callers and integrating across callers across different groups, sort of with a common thematic approach, which again, it's something we can essentially just take on in the context, especially of what Joe just mentioned as an opportunity.
Well, I really think that should come from, as a directive from the NIH, because the individual sequencing centers are going to apply whatever they gravitate towards, and it's often something that they wrote. But you know who's going to suffer is us, because no one wants to recall SVs from genomes. It's a huge undertaking. So I would just recommend, if it's possible, to try to get the sequencing centers talking and to arrive at a best practices. It might just be GATK, but even within GATK, you know, there are methods that can vary quite a bit. And if we're going to have, if somebody's sequencing theirs at, forget all the different sequencing centers, Hudson Alpha or something like this, and another person's at Broad, you really cannot compare SV between studies unless they're really all done uniformly.
Right. And again, you know, I think to your point, Joe, and you know, I don't know what your take is on Joe's comment, Valerie, but you know, that's the other role that, you know, recognizing these challenges that emerge across sequencing centers, but recognizing that there, as far as we can tell, there'll be more than one sequencing center for the remainder of this time, and also in many other programs. You know, that there are opportunities to evaluate, you know, this being as part of the sort of DRC or DCC like functionality in whatever effort, which is certainly, you know, I think it's something we saw as a opportunity and potentially a responsibility on the DRC side, for example, to implement. And then this also will allow you to do some of the things that you're talking about, which is bring in your own cohort or other data sets and harmonize them with Kids First data sets if there's a common workflow that can be implemented.
This is Ali. I was wondering how people envision using additional data set. Is it, you know, finding good genes in their own cohort and doing lookups in other data sets? because there are also opportunities to deliberately combine them and do scans across the genome, collapsing-based tests, looking for even rare or common variants together. And I wonder if we should deliberately think about some of these based on existing, overlap between phenotypes or genetic architecture.
I agree with you, Ali. I think the future is going to be collapsing analysis and statistical analysis, and that's going to require more and more harmonization of data.
That's right.
That's going to be ideally to the point of joint calling. It might not be practical to do joint calling, but at least to make sure as much as possible between the two or whatever number of different sequencing centers that we lock down a methodology.
And I think, we have trios for the most part. So at some point we'll have to think about controls as well for, again, case control analyses if needed, if the data sets get enlarged, or there are additional data sets that can be brought in.
Yeah, that was going to be my question also. I mean, is there any discussion on importing large control cohorts that we can harmonize and have analyzed with a consistent pipeline that are non-cancer or structural birth defect?
Yes, I think this is what I was trying to sort of hint at earlier is not everybody agrees what's the perfect control as a cohort. And so I think this is where it'd be great to get some input on this. And we have opportunities, especially through the intersection of the Kids First efforts with other Common Fund supported data sets. But for example, it's not clear if everybody would agree that GTEx genomes or expression data are the best controls, or given a lack of age match, for example. But these are the kind of questions that if you have identified or think that there are good cohorts for controls, we'd love to hear about that. And then the alternative would also be, sort of cross data set controls, as I think was mentioned a couple of times here.
Right. Yeah, we've thought about that also, but I just think it's going to be a really important thing to address. So for some of our studies, we've reached out, for example, locally to the PenMed Biobank, and we're, have a proposal to analyze those data as controls, and they're continuing to grow in terms of the DNA, not so much the RNA, but I just think if we can find control cohorts that were generated at one institution or at least company, that would help also.
Yeah.
I think another opportunity is that since we have trios and presumably a number of parents are going to be healthy, it'd be very good to have as much, sort of information about their health and the clinical conditions. Very often, so the kids are really severely affected, but there isn't as much of a phenotypic sort of evaluation in parents. And if we do have such data, we might be able to use at least a subset as controls, at least to get us started.
Right.
To be a little careful with that, because in some of the cohorts I've analyzed, the parents do carry some kind of background burden.
Sure.
No doubt. But we have the sequence and we have them in the data set. And so here we can, I don't know how much of an effort it is to actually go back and evaluate the parents. And we still may not be able to clean that up because of non-penetrance or because of just there's a genetic background effect.
Yeah. And some of this could be, phenotypically driven, right? And I think, I like the idea that, I think it might have been who you mentioned, right? While you can approach this sort of holistically and say, okay, we want to do, let's say, covariant calling on all cohorts or joint calling on all cohorts. I wonder if there is, the opportunity to really be data-driven about what genomic analyses we want to do at larger scale based on phenotypic annotation, or, let's say to the parental control context, you can take example of the most divergent cohorts. Typically the parents of those cohorts could act as controls for each other, or any of those kind of combinations of selection that might make sense for the group. And again, yeah, I think, what you're hearing, at least from us at the DRC and also from, I think, Valerie and the NH, is that there really is an opportunity for this to be community-driven and informed in the way that we structure some of these efforts.
Get some deeper phenotyping as well in some of the cohorts, especially the large cohorts, maybe the orofacial cleft cohorts. If we have an opportunity to get more data from face space or something like that, you could select your controls based on, as Adam was just talking about, good separation between phenotype groups. Just looking at how well they separate on a phenotype map. And that could be one way to explore it. But, trio analysis is also, if you do a good trio analysis, you can try to do without controls by the fact that you do have trios. And then, of course, when you have the quads, that makes it even easier. There's also ways of putting together artificial controls and other ways to try to game it. Doesn't mean it's perfect, but we do have a lot of patients in this cohort. We could do virtual mating of different genomes and create controls. People have done that in the past. So just things to think about.
I like that this one question sparked a lot of really great input. These themes are not new, but it's been a little while, so I'm glad to revisit them. Thanks so much. Maybe we should keep going down the list.
Yeah, sounds great. So our next question is, what challenges have you faced when accessing or analyzing phenotypes in other studies? Someone already responded with dependencies on the wide variety of formats, whether it be PDF format or docs or other medical records. So kind of like a lack of standardized format, sounds like. If anyone else has any other thoughts on this, feel free to type them in or just say them out loud.
Well, I'm going to add what Dr. Gharavi raised, which is lack of confirmed information about the parents, or lack of confirmed health information about the parents.
I think about the parents regarding de novos, I think there's always a risk that a parent is a carrier for the de novo, but just not penetrant, or maybe there's some other variant in there, as we've heard from some of the speakers today. But for recessives, definitely the parent's a carrier, but definitely it does not have the second copy of the mutation. So they might be considered better controls. The problem is we're going to suffer from population stratification, because those are largely coming from special populations. So I'm not sure we're going to be able to solve the parent, be able to use the parents that effectively as controls. I'd be a little concerned about it. But I mean, I think for our study, at least, I would feel comfortable using a neuroblastoma affected cohort as a control for collapsing analysis for something that we're doing in general, because the diseases are pretty non-overlapping. But if it was, let's say, cleft lip and palate or something like that for our neural tube defects, there might be enough overlap that I'd be worried that we might run into the same genes.
That's a good point.
So are we, I haven't really done a lookup on other data sets, so I'm just going to ask this question. Are we using the same terminology to describe the phenotypes that we're all interested in across data sets? So for example, if I'm interested in somebody who has, let's say, a kidney malformation, hypoplasia, cystic kidney disease, and so forth, are these sort of represented in some way? That are we all talking about the same thing? Often I find there's a checkbox against kidney problems, but those could be multiple different issues.
It depends on what comes out of the clinical description from the originating investigator.
Right.
So we do our best to cast, we talk to the investigator if we don't get HPO terms from them. Nicole and I we actually, Nicole spends time to go through a lot of the terms that aren't well defined. And if it's not defined, it goes into HPO. So we try to use HPO, NCIT, Uberon, and Mondo to try to define terms and diseases on the same playing field. Some cohorts have very high level definitions. For example, if the child has a birth defect, they'll just indicate CHD, congenital heart defect, but they won't go into detail. I think that's the CDH cohort does that. So we have different levels of specificity, and that's something that is out of the investigator, and sometimes the limitations of their study means they can't go back and better characterize that data. So so there may be more information that's just lost. But when we do get very specific terms, we try to be as specific as possible with at least the HPO and the other terminology ontologies.
And so I think to your answer, the answer is, I wasn't sure when you said, are we all using? I wasn't sure who the we was when you're asking that question. But, as Deanne's pointing to, there's remarkable heterogeneity in that process. And it's something that we don't have a perfect answer for. Alison will do a brief demo of some of the way we've been tackling this, and paired with the effort that Deanne and Nicole undertake to harmonize the data, we also try to make the source data or annotation available for the cohorts. And there are a variety of different ways to try and solve this problem, including forcing submitters to align to one or the other ontology. But then there are costs of trying to force investigators to do some of those things. And this is some of the reasons that I think we're trying to really iterate on how source data of various formats can come into the platform in a more routine and easy manner. But we're always open for input on making that easier, which I think is partly why this question is here.
So I think this is great that you're doing all the combining this fiction, the HPO terms and so forth. I guess if there are joint analyses that are planned, then there may be ways of then for the individual investigators to try to harmonize the phenotypes. Alternatively, if there are certain traits that we are sort of all interested in, and certain information that we're all interested in, that might be something we can jointly decide to try to look for and put in that effort so that everybody benefits.
Yes, I agree.
I mean, some of the stuff is maybe just the age at which the sample was collected, for example, those kinds of things that might, if somebody wants to look at mosaicism or somatic mutations and try to harmonize this also to their own data sets, that may be helpful.
Let's keep moving through so that Alison has some time for her demo, and then Adam has time for his talk.
Sure. Okay. Our next question is in the purple box. What is your preferred format for receiving clinical data?
I'm going to say spreadsheets just to be inflammatory.
Perfect. Word docs, PDFs. Oh yeah. Anybody like it? Any kind of computational formats, like think by email?
How about something structured that we can import and export into our own preferred programs, data sets?
Yeah, what would be a good example of something structured?
Oh, you're talking to somebody who doesn't really do a lot of the stuff. I just asked others to do this for me. Let me find out.
Well, yeah, I'm going to say something from FHIR, ? But this is very computational on the back end, and Adam and Alison always yell at me that on the front end, nobody will know it's FHIR. But I'm going to put it in.
I mean, that's a good point, Valerie. I mean, I think we talked to a lot of the bioinformaticians on this, and I think there is a difference between, wanting to review kind of patient cases or things that are complex and doing really large scale analyses. And I think there's some tension there sometimes as well. So, I mean, it would be good to hear about, like, in your past studies or other things you've done. I mean, if it's just practically like doing spreadsheets or, is it ultimately going into some R Python code, or are you really looking at kind of individual patient cases in detail?
Right, and some differences just that arise also, whether it's sort of a representation of a, per patient case report form in a spreadsheet versus, summary data in a matrix of some sort. Whether you like to get, clinical data paired with genomic data, or you do that separately. But to Allison's point, it just, I think, often depends on what kind of analyst or hypothesis generator you are.
It's okay if we're a little stuck here, and people can keep filling this out, and we'll save the link.
Yeah. All right. So the next question is, oh, we're ready to go on? What tools do you currently use to support your phenotypic data efforts for data collection, data annotation, analysis, etc.?
Seeing boots come in for Red Cat.
I don't know, Sharon or Phil, when it comes to interacting, let's say, with the COG-based data sets, do you guys actually interact with the sort of metadata array rule, or down much further stream than that on any of the clinical data side?
So from my experience, it's typically just been a COG statistician providing an Excel spreadsheet with the variables of interest by COG IDs. I've not personally downloaded data directly from Rave. I assume that the statisticians at COG probably do that. I don't know about Sharon's experience.
Yeah, this is Sharon. I agree. That's been our experience as well. We typically just receive an Excel spreadsheet and then we import those data into a local database.
Okay.
We've used some structured PDF files that investigators can fill out, and then it just emails it directly to us, and we can import it into RedCap. It takes a little bit of work up front, but it makes it much easier on the other end.
Cool. Sounds good. The next question is, what tools or processes would help support your phenotypic data efforts in the Kids First data resource? So things that can help you with data curation, data harmonization, data analysis. So any kind of tools or workflows that could aid in this process. Maybe something that might assist with mapping clinical terminology to HBO or Mondo or other ontology terms. I think those kinds of things are things that we're looking for.
Yeah, and I'm trying to think about what would, back to Dr. Gharavi's point about, honing in on sets of phenotypes that clearly are of interest across groups. I think we've already identified heart defects and some other examples. But what is the best way to actually start tackling that, other than us? Maybe it's just about getting together and having some meetings and going over some of these structured data sets.
Well, I think in part it has to be driven by the data sets that we have, the number of different samples, and the science that's going to drive this in terms of the collaborations that might evolve out of this. So that might be something worth talking about, whether in the context of another meeting or again subsequent to this meeting, if there are individuals who want to work together specifically on trying to do a cross phenotype analysis, that would be good.
Yeah.
I mean, for example, sorry. I was going to say, for example, we have a bunch of patients who have, structural brain defects in addition to their kidney phenotypes. So those obviously would be great candidates to study with, other structural brain defects as well. And so, and if there are, obviously we'd be very interested in any of the patients who also have some sort of congenital kidney defects to be able to do a cross phenotype analysis. I think those are the kind of things that make sense. And we're open for business that way. That would make sense. That we can try to build cohorts this way.
Would it help, do you think, if sort of on a DRC side, we identified, quote unquote, cross disease cohorts based on phenotype and proposed some of these to you? Sort of highlight for you a priori,
Yes, absolutely.
Yeah.
Absolutely. Yeah. I mean, we can each give you our our HPO terms of interest, for example.
Okay.
I'm curious to know who just put up the comment about model systems. This is certainly on our radar. Well, anyway, you don't have to reveal yourself, but we'll talk about it.
Sounds good. Should we move on to the next one?
Sure.
So are any of you using common data elements? And if so, which ones?
Can you just explain what that is for people that might not know?
So a common data element is like a standardized data element that might be used for things like collecting demographic information or like gender information. So like gender would be like male, female versus boy, girl, something like that. They're developed by I don't know if exclusively, but there's a bunch of common data elements developed by the NIH that can be used for like survey instruments and other kinds of data collection instruments.
Adam, Allison, or team, this is Jamie. I'm just curious because we've talked about this a lot recently in cancer and for what we're doing for CCDI. So relative to COG's, say 500 some CDEs for Project Every Child, how do the structural birth defects data line up with that in your experience at Kids First?
I think it's actually, I'll let Allison and Deanne answer first before I give my perception.
Yeah, I mean, I don't think we've necessarily gone through those 500 CDEs. We've done a lot intersecting, I think, GDC with a lot of the structural birth defects. And I think it is interesting. I do find that on the structural birth defects, you hear people talking much more about HPO and the phenotypes and different kind of survey instruments. And so, I think it's a good question, like how common are common data elements? I think often kind of the demographics and things like that, yes, people tend to reuse the common data elements, but as you get more and more to the different disease specific, even at the level of cancer and structural birth defects, you start seeing common data elements that are common for that scope or domain. And I think it's something that we've been talking a lot about. And what I think, we even heard it earlier when we were talking about how do we co-analyze even the cancer and structural birth defect with different linkages of those data elements to different genes. And so I think it's an ongoing process. And actually it would actually be, I think, a good project actually to go through the Project Every Child CDEs and kind of ask that question in a more fine-grained way. But I'll let Adam or Deanne also address it.
I agree with Allison, and I think because of the domain specific, the way the data is collected for each domain, it does make it really hard to overlap. Often people don't, it depends on the project, but I'm not sure if people record a lot of same types of phenotypes in each domain. So that's why in our common data elements ourselves, we've had to kind of elucidate which are cancer specific and which aren't. So
I guess,
yeah.
Oh, sorry.
No, go ahead.
I was just going to say that just remind, I think that's why we're talking a lot about these ontologies and stuff, because the power of that is instead of a common data element, you really put this into a knowledge graph where someone's checking, kidney disorder, that you can maybe, follow up, or if you find interesting findings, kind of do finer grained phenotyping. And I'll kind of show a little bit of this in our demo of how we're trying to get to a bit of this in the portal, intersecting those. And so I think that's why, CDEs asking that first question of how they align is important. But it's really the next level of how does that map into places that are going to allow you to move up and down in terms of how you're thinking about these diseases and phenotypes.
And my impression is exactly what Allison and Deanne just mentioned, and I put the link, to the NLM sort of resource of common data elements. My sense that very few people actually look a priori before they're designing a study at such a resource, but I might be wrong to look if there are existing common data elements that they should be implementing or not. But again, maybe that's biased.
I guess I would say that's actually probably a cancer bias.
Maybe that's true.
Because I think often if you're doing lifestyle surveys or you're doing
that's fair.
the these are often like used. That's been my experience actually, kind of coming from more on the cancer side and moving more into this kind of cross disease world.
And there's the obvious needs for pediatric. A lot of this comes down to the fact of pediatric diseases being different than adults as well, which was kind of following with Allison was saying. That's also its own domain. So what we're looking for phenotypic analysis in children is going to be different than adults. So we may need to look at, we may need to add, like what HBO is doing, a lot of very specific terms and concepts going forward. And I also want to use this as a plug for FHIR, because if you can get a holistic medical record on a child, you start, the data elements can still be put into common data element type slots, but you get a more comprehensive view of the profile rather than, because a lot of people, when they're trying to come up with the idea of a data element for a cohort, I found that in the Kids First data, it is very limited, right? Because they're only studying certain things. So it would be really neat to eventually be able to get a more holistic view of the profile of the subject. Because then that starts to get around the idea of these silos of specific types of elements, and it opens it up a little bit more. But that's just my 2 cents. Other people may disagree.
Okay, I think the meat of these questions is really about there's the last 3 questions, but I think these are supposed to be 2 because the first one is a yes/no question. But we really want to know which data elements you would want to obtain if you could go back, based on the talks you heard today or the talks you heard in the first webinar. And then, which elements would you go back and collect for your cohort? And then which data elements would you want everyone else to collect so that, for example, you could cross-analyze that with your cohort?
Well, people are thinking about that. This is John Hartman. I just couldn't find my microphone button, but I was the experimental model systems question guy. So I was being shy. It was just that I couldn't find my voice.
So I'm going to go ahead and add, just back to Dr. Gharavi's point about the parents, if there was an opportunity to go back and confirm some things about the parent, maybe that would be worthwhile, whether they were, I think congenital diaphragmatic hernia is an example where there might be low penetrance, but if you look closely at family members who didn't have any symptoms, they might actually have some diaphragm abnormalities if you did an X-ray.
And I was interested in Sharon's talk where you talked, Sharon, about almost essentially exposure data in pregnancy or childhood, if that's a common data element or question that people would be interested or not.
Hi, this is Sharon. I'm actually not aware if that is a common data element or not. I'd be interested to know, and I'd be interested to know if other people would be interested in those data as well.
Oh, and sorry, for this question, it doesn't have to be common. It's whatever you would dream of that you would want.
Sorry.
So one thing to
answer
the
talking. This is Jamie. I was just curious which one Sharon was asking for, but sorry.
Oh, I think Sharon was describing, I think it was I forget which study, it was a new study that you were talking about, Sharon, right? That you're going to be collecting or already have that data as it relates to smoking, food, other things.
Yeah. So the neuroblastoma X01 cohort was based off of the NINA study that Andy Olshan ran at UNC. So he basically built a questionnaire and collected a lot of data on exposure during pregnancy, which is, I think, what Adam's asking about in terms of like tobacco usage, vitamin usage, diet, what you drank, things like that. You know, with the goal of trying to understand if that might have impacted, obviously, the development of neuroblastoma in our case. But I think what Adam's asking about is whether other groups might find data such like that useful to evaluate their cohorts as well. Right?
Got it. And we do have common data elements around exposure, at least as far as being on the list, if that was the question. But
okay.
I just wanted to make sure I answered it because I got pulled away for a second. Sorry about that.
I think the PCGC may also have, and they may have not reported it, but it's probably available, some information on the maternal smoking and things like that related to the heart defect cohort, just for the record.
I thought I heard Phil maybe chime in earlier, but
yeah, I was going to say, at least for the cancer cohorts, I know that the questionnaires for the Ewing sarcoma, neuroblastoma, and rhabdomyosarcoma cohorts aren't identical. They were largely conceived in the COG epidemiology committee, so there's probably a lot of overlap in terms of questions that were asked of each of those trio data sets. So that would be something to consider again if we're considering those exposures as part of the CDEs, and it sounded like Jamie said they are in fact. So just again, something that we can talk about, this, you know, adding and characterizing a bit better.
Great.
I think this is great. I think it's okay that there's some shyness, and I think people should feel free to keep adding thoughts here. It'll be really useful for us to have this documented and to revisit for you know, our roadmap moving forward. Allison, I think we should turn it over to you. Are you ready for some portal demo? You're muted. I don't know.
One more thing I would add is anything about maternal the gestation. I don't know if that's included in what you're saying, but for so many of us looking at developmental disorders, any history of complications during gestations, you said parental history, but maternal history of diabetes, for example, is really important for kidney malformations and other, I'm assuming, other disorders. So those kinds of simple things that might be of interest to.
Yeah, I agree. And that was collected through the neuroblastoma study. There's data.
Yeah, I think it's definitely a recurring theme we've been hearing. So if other folks or specific aspects about it too would be great. You know, feel free, I think, really to use that board if you want to put specific things down. Think of it kind of as maybe a wish list a little bit that we can keep together. So I'm gonna go through this pretty quickly and then hand it over to Adam a little bit. But I did want to talk a little bit about the portal. So I hope everyone here has actually logged in at least once. If you haven't, definitely highly encourage you to do that. And you know, over the last couple of months, we've released actually a lot of data. So we're very close to reaching the 15,000 participant threshold and close to 5,000 different families now. And so I think a lot of you will get emails pretty soon. We've been focusing very hard on the variant side. So we're going to be rolling out the variant database over the next couple of months. Initially with folks who have their data in hand, we'll be doing a beta rollout and then kind of more widely to the public. But, you know, since today has really been about phenotypic data, I wanted to, you know, highlight just some things that we've been doing in terms of trying to create these virtual cohorts. And a lot of what we're doing here, gathering this information, you know, some of it is about kind of the data and the NIH side and what can you help resource to collect. But I think from the DRC side, it's also been, we're going to roll out the variant database. What are the next kind of early next year? We can really start road mapping, some of these clinical features that we're talking about. So if you haven't been paying attention, it'd probably be pretty easy to miss, but we've added a new feature under clinical here to be able to browse the HPO phenotype. So here what you're seeing is all of those participants ones that have annotations with HPO, and a tree has been filtered to say, here is on this column here, all the participants that have been annotated with this specific HPO terminology, and then here is all the patients that have something underneath that. So you can see, cardiovascular system, which was a theme today. You can see at the very high level, we actually have 41 participants. So these are probably ones that aren't necessarily focused on heart disease, just annotating there's some cardiovascular issue. And then you can start seeing the subgroupings of, how many are in the different morphologies, and start really digging down into, okay, so yes, probably in, maybe PCGC or other ones, there's been a very fine grained phenotyping here. And you can see at this term, there's 130 participants. So there's one way of kind of browsing the tree and looking at it. You can also type in, whatever, kind of different things you want to look at. Let me think of, not very good at spelling on the fly, but you can look at, search this and it'll highlight the different terms that have diaphragm. And similarly, you can see these numbers of, how many were phenotyped up here, how many were phenotyped at a lower level. And what you can do once you do that, instead of kind of going to a specific term, you could select one that's higher up, add that, and I move my window, apply it, and that will bring it into the browser and immediately see, yes, there's a lot of the diaphragmatic hernia, there's a few with congenital heart disease and the pediatric brain tumor atlas. Another one, and if you haven't really used this before, what you can actually do is start new queries, so you can keep that one. But if you're interested in another phenotype, we can go back here again, and let's see, I know one of them. So you can see that all of those are at that level. You can see this phenotype is actually across multiple different studies. So you have the congenital heart disease, but also the cranial dysnervation, some of the brain tumor, and some of the diaphragmatic hernia. And then you can see from this phenotype that here's the kind of most frequent diagnosis for that. So just kind of at a glance, being able to see those, and then you can actually combine this in different ways. So if you wanted to or those together and see everyone who has one or the other phenotype, or you could even combine them and try to say the ones that have both of those phenotypes. You can see that there's only a handful here, but these could be of interest. And something I'll also show, you can actually switch to table view and see the actual participants and see the different diagnoses that they have, the different files associated. And then, look at them at the participant level. So for example, if we look at this congenital heart defect one, we can see the different specimens, the type of data that was generated. And then, this is part of what I was alluding to a little bit about if we do have richer clinical data, what kind of views, you can see that this participant's been, has multiple different diagnoses, multiple different phenotypes. We can support ages, maybe longitudinal views of this, if that would be available for certain types of diseases. So these are some of the things that we're really trying to brainstorm and think about what kind of tools and views and how can we make this kind of really supporting some of that discovery process and, understanding, kind of what we talked about today kind of there. And, there's a whole other slew of features in the portal. I'm not going to go over it today. I really just want to focus on that. I will note that we've rolled it out for HPO. We are now working on ones that don't have. the kind of, this is the old view of how the search and filtering was done. So hopefully this new view makes the ontologies a lot more useful, and then we'll be rolling them out for NCIT, Mondo, and I think we actually have Uberon as well for some of the anatomical sites. So this is kind of our first foray into it. So I'd love to get any feedback, thoughts, if that sparks any new cool ideas of what you think we could do with some of this data. We really want to make it useful to folks. And we, the more we can make these tools link to the standards that we're recommending, and not just kind of recommending standards for standard's sake, but really, if you do it here or you do use these ontologies, one, those ontologies will grow, they'll support your disease system better, but then it can also create powerful tools for your analysis as well. So I just wanted to show some practical demos. I really encourage you to go in there and check it out, or have people in your lab go and do it. We're always interested in feedback for sure.
And just to Allison's point, our hope is that these tools can, in part, address some of the challenges that Deanne highlighted, where you'll have cohorts annotated at sort of different levels of ontology. But then really, as Allison hinted, many of the Exo-1 investigators will get emails later on about, how can those types of cohort building activities really intersect with your own cohort's queryability at the variant warehouse level and the tools that are being deployed for variant analysis. So we're super eager sort of getting feedback about how we can empower you as you move forward, initially the variant context, but then, as Joe and others mentioned, moving into structural variations and, other methodologies.
This is Joe. Can I ask you, Allison, how, or maybe I can ask some of the groups that have been already depositing sequence, how have you been using these kinds of terms as you evaluate your genomes? Like, I'm used to using software where it incorporates both phenotypes and genotypes together, but have people found it useful just to have a really rich phenotype database without having the genotypes integrated?
I think it's a great question, Joe, and while people are thinking about that answer, one of the things is, and I hinted at this, it's one of the other questions on whether people like to see them both together. And like Hugh, I think there is a bias to be sort of seeing an integration, at least at some level. We hope this can be done on a variant warehouse side, but one of the things that we're also trying to navigate in the portal is the capacity of investigators to query phenotypes before having to go through the dbGaP application process. So really trying to sort of understand whether through a cohort building activity at the phenotype level, then they can sort of discover that they should apply for the controlled access genotype level, for other cohorts and do such analyses. Because, and this is true for myself as well, right? We all initially begin with the known space within our own disease of interest initially. But that's partly why you see the segregation of the tools in the portal to this. But in the variant warehouse space, you'll be able to do both types of analyses together across both genotypes and phenotypes. Does that make sense? Yep, so sorry, thanks. So I'll just take a couple minutes, just really, this has been a phenomenal webinar. Thanks so much for the presenters, and especially for all the feedback and ideas. I think we'll regroup with a lot of next steps. But I also wanted to highlight some of the next steps we're taking from an infrastructure and DRC perspective. And some of it has to do with really expanding this type-to-type analysis and really thinking about additional data modalities. And I've had a chance to speak to several of you regarding this, recognizing that in different cohorts, and I think Valerie hinted at this earlier too, there are other data modalities that people incorporate into their analyses. And we've thought about this to some extent, and you'll find, for example, if you go through the portal, that we've included other data modalities already in the file browser. But each data modality requires an additional set of tools to truly empower it for cohort building or integration for downstream analyses. One of the first ones we're taking on is really imaging data modalities. So in the very near future, we'll start integrating these tools in ways that then allow DICOM-based images, at least in this context, although the tools will also support JPEG or other image file types and their integration into it. But this will allow you to really deconvolute, what just says DCM as a file, but really look at all the different sessions that, are incorporated in a DICOM image in that context. And again, super eager to really hear about use cases that people are engaging as it relates to the integration of imaging plus genotypes or imaging plus phenotypes as part of their workflows. Timelines. I often hinted at this, and we always talk about time as a variable that we try to optimize in Kids First because our mission is to accelerate research. But I was really thinking about another context of time, and that has to do with the earlier part of the mission, which talks about our mission to alleviate suffering. And almost by definition, suffering is sort of pain over a quotient of time, and our job is to alleviate that. And time as a variable in the data set is actually something that's been woefully missing from many of our studies, with capacity to capture longitudinality of data, not only to sort of capture, whether multiple phenotypes occur across time in particular subjects or the way that they are presented, but also, when we think about even cancer, it's not uncommon for a cancer patient to have multiple recurrences or presentations. And again, that's another element of time. So we really are focusing on, how do you capture, represent, and support time-based analytics in ways that can empower the discovery context. And part of that is really to integrate with outcomes, because obviously that's really the total mission is to change the outcomes for these children. And so as Valerie hinted, and I think as Allison and her team have really been doing an amazing job, one of the ways that we are trying to leverage the FHIR-based architecture that's really being integrated into the EHR context is to think about longitudinality, as Deanne mentioned, more comprehensive representation of patients, perhaps accelerated ingest of data, and really evolving the FHIR-based architecture to support research-based data sets, which then can be integrated or evolve into other data models, but really to provide a much more robust architecture for the ingest of data from diverse sources, recognizing that EHRs are really one routine source that many of us use, plus then the integration of other REDCap or questionnaires or other instruments. So our hope is to really leverage some of the Kids First cohorts as initial use cases, and then really iterate on this infrastructure more broadly, and hopefully empower longitudinal and more comprehensive clinical and phenotypic data integration into the cohorts. So that's really some of the sort of upcoming features and work that we'll be doing on the DRC side. And then I'll turn it back over to Valerie for a few more comments.
Yeah, we hinted at this today as well, this theme of enabling cross data set analysis across model organism databases or data sets, and across the human whole genome data sets is really important. And I think we're really curious to get more thoughts and input on this to really drive how we could intersect. I think we've identified COMP2, but also zebrafish and Xenopus as models that are heavily used by this community. So we're just going to go ahead and tackle this head on for part 3, which has not been scheduled yet. We will be in touch. I'm pretty aware that a few of you have worked with COMP2, have made your own zebrafish models. If you are interested, if you want to be a speaker, like today, to talk about model organism work for the part 3, please let me know. But I do have a few of you on my list who I will approach. But what we're also going to talk about this on the database level, how to enable the computational analysis to then do the functional genomics in a model system. And I think there's a lot of opportunity here, but we need your help to define that roadmap. Okay, well, with that, we're a little bit over time, but it wasn't too bad. Thank you all so much. I think this was really helpful for me. It was really great. I think we get some really great ideas from you, the experts, like I said at the beginning. And, we'll be in touch when it's time to schedule the part 3 webinar. But I think there potentially is this place where we might start forming working groups around some of the specific questions, specific phenotypes. And we'll take that back to the DRC and think about ways to support that as well.
Thanks so much.
Thank you.
Thank you. This was great.
Thanks, everyone.
Thanks so much, everyone. Take care. Bye.